Hello, ladies and gentlemen, colleagues. Good evening to everyone. We're going to start off our Plenary 5 tonight. And to kick us off and to start us off tonight, I'd like to call on the chair of the next session, Dr. Evelina Lagamayo, to introduce our speaker.
Good evening. I'm happy to introduce to you our speaker for tonight. Our speaker for tonight is a molecular microbiologist and an infectious disease specialist. He took his bachelor's degree in microbiology at the University of Santo Tomas and his medical degree and specialty in internal medicine at the LaSalle Health Sciences Institute. He is the Deputy Executive Director of the Philippine Genome Center at the University of the Philippines System, the Director of the National Training Center for Biosafety and Biosecurity, National Institute of Health, University of the Philippines, Manila. He is the recipient of multiple national and international awards, and most important achievement is the Lab in a Mug project, with its principal product on the biotech MTM dengue kit. Okay. The latest project is about the local RT-PCR, which is used for the detection of the SARS-CoV-2 virus. Ladies and gentlemen, let us welcome Dr. Raul Destura to give us a lecture on the navigating the maze of COVID-19 diagnostics.
Thank you very much, Dr. Lagamayo, for the kind introduction. And I would like to express my gratitude to the organizing committee of the Philippine Society for Microbiology and Infectious Disease on its gold year as a society. And I am very proud to be a part of this organization as well. And this is my hope to contribute to this fantastic event in a way I can on the topic navigating the maze of COVID-19 diagnostics. But before I proceed, I would like to make my disclosures. I'm the founder and CEO of the Manila Health Tech Incorporated and the principal developer of the local COVID-19 test kits, the Gen Amplify RT-PCR detection kit, and the Saliva Fast Direct RT-PCR technology. So the outline of my talk involves some overview of COVID-19, but we'll just give a snapshot because I'm sure the audience has already received a lot of information on the local and global perspective of COVID-19. A little bit on the issues on viral dynamics and timing of the diagnostics, because some of the most common questions I always receive from colleagues almost on a daily basis. Some inputs on serological testing, molecular diagnostics, the use of artificial intelligence and image recognition software, what are the existing sequencing platforms dedicated for COVID testing, a few discussions on some unmet needs in the current diagnostics and what is actually emerging out there that we can all look forward to, and some concluding remark from myself. So as of yesterday, this is the heat map of the global situation of the COVID-19 pandemic. There are about over 62 million confirmed cases and about close to 1.5 million deaths reported worldwide. And based on the prevalences, it ranks from the Americas down to Europe, South Asia, the Eastern Mediterranean, Africa, and then Western Pacific in terms of the number of confirmed cases reported to the global data. As of this morning, out of more than close to 5.5 million tested here in the Philippines, there is a record of 432,925 cases seen. But if you look at the curve, we're seeing some downward trend of number of positive cases, but We have to agree with what is our Department of Health keeps on telling the public about being cautious about this downward trend, because we cannot relax at this point in terms of our mechanisms for controlling disease transmission, principally on physical distancing and the wearing of PPEs or face masks to further reduce the risk of transmission to our countrymen. So we cannot overemphasize the use of physical means in controlling the disease while we are waiting for the definitive intervention wherein vaccines to arrive hopefully in the Philippines, and only at the time wherein when we see the flip
Pati yung
between
mga
positive
upcoming.
cases
Kay Dr.
flip between positive cases and the total number of vaccine coverage, we can actually see a good herd immunity in the control of the epidemic. But what is that ratio still needs to be seen, but there are several mathematical models in previous epidemics ranging from 40% to 95% coverage to flip the effects of the transmission dynamics of the coronavirus. So it's still a lot of learning along the way with this new virus, with the global scientists and researchers and clinicians are contributing to this information. There is currently a very high demand for COVID-19 diagnostic due to the still increasing number of new cases and the continuous global epidemiological surveillance. And that if you look at the viral dynamics of the SARS-CoV virus, the challenge here is really identifying the pre-symptomatics that can actually cause transmission of the virus before they actually know that they are already infectious. In this particular table, this particular graph, you will actually see that before a patient develops symptom, there is already an inoculum size enough to infect an individual several days before the onset of symptoms. And in this particular graph, it nicely describes where our current technologies can actually be utilized depending on what purpose. If your purpose is active case detection, the most likely technology that you will use is the molecular-based tools, such as polymerase chain reaction, because it has the earliest detectable power, as opposed to antibody detections, which you see its power towards the end of the second week, picking up on the third week. But it depends on what your question is. If you are determining active case findings, then PCR detection is the useful tool. But if you are more interested on the degree of exposures in the community, perhaps an antibody detection test can be a good alternative. Now, but not for active case finding. In looking at the viral infections among asymptomatics, there are actually a viral transmission in terms of symptomatic individuals. There's still really no accepted proportion of asymptomatic COVID-19 patients globally. In this particular review, it estimates around 20% out of all those infected can actually cause disease transmission. And the risk of transmission is at 0.35. But if you look at the plot here, it's really, really wide to determine which one really is, how high really is the transmission risk for symptomatics. Having said that, it's always best to err on the safety caution, which means I would still recommend social physical distancing and the wearing of PPEs for those who tested positive despite the absence of symptoms. And it's really wide, from 6% to 60%, 69%, depending on which studies you are actually citing, for a symptomatic contribution to infection. This wide gap gives us an information that we cannot relax in terms of symptomatic individuals testing positive. But it's easier said than done, because sometimes when a person who is asymptomatic based on experience tested positive, and then we look for another laboratory to check and confirm, and then they turn positive on the other one, and then they would believe the negative one and not the positive result, which this really destroys the purpose of the testing in itself. If you look at the viral kinetics in the symptomatic individual, to your left side is the cycling threshold. When you see this, when you say cycling threshold, this is the period at which the start of the logarithmic amplification of the gene targets in the PCR cycle. And this one describes two types of genes that are commonly used in PCR kits. On the left panel is the envelope gene. On the right panel is the RNA-dependent RNA polymerase RDRP gene, which is confined within the open reading frame 1A and B genes for the coronavirus, for the COVID-19 virus. If you notice in this plots, majority of the cycling thresholds for asymptomatic individuals falls within the city value between 30 onwards, higher. The higher the city value, the lower is the virological burden. It's a semi-quantitative assessment of the virus in terms of the inoculum size. But as to whether this inoculum size is enough to transmit, or is this a viral product, or is this a whole virus that is actually still viable for infection, still remains to be continually studied for us to understand how far should interpretations of the cycling threshold values in determining whether the disease is just viral shedding of a non-infectious viral proteins, or it's really a whole viable infectious virus that's still being shed. Take note of the variability as well in terms of which type of gene targets they are actually studying. If you look at in this study by Lee, previously just released this year, in terms of the conversion rates, conversion meaning from positive to a negative PCR, there is no significant difference between asymptomatic and symptomatic, including pre-symptomatics. And the positivity rate can go beyond day 28 of the diagnosis. But whether that particular positivity rate is infectious or not, I really wish we have that very robust data to establish, but the design is very problematic. You will need a biosafety, at least biosafety level 3 laboratories to do viral culture studies for the TCID or the median median tissue culture infectious dose, or the TCID 50 in this case. In terms of the viral shedding, for symptomatic or post fully established in much bigger and well-designed studies. Now, there is an interesting paper that describes infectious dose in terms of the city value. I kept on mentioning the city value because the most common platform for the diagnosis of coronavirus is the PCR test. And the diagnosis is based on the city threshold and depends on which type of kits you are using. Some kits are more strict having a cutoff of 38, 35 to 38, others 30. Others are more lenient, having a cutoff of 40 because it's all under emergency use license and unverifiable value, which one really is significantly infectious or not. But it's a good thing because of this variation, people are able to identify variations in the cycling threshold. In this case, for example, when samples collected and inoculated in a tissue culture setup, CT values that goes beyond 24 are no longer showing infectiousness on the viral culture. There is no plaque reductions and all of these things when they measure the median tissue culture infectious dose, or the TCID 50. But there is a caveat to this. Take note that when the study was done, this was based on a CT cycling threshold of a single gene target. I think in this case was an S gene or an N gene. The viral dynamics of targeting genes for PCR varies from which gene targets you have in terms of the robustness. When the SARS coronavirus was isolated from the lower respiratory tract specimen, all of the diagnostic tests around the world developed using the RT-PCR platform. And the gene targets for which these PCR kits were done are based on the following genes: the RDRP DNA RNA polymerase gene of the ORF1AB sequence, the E gene, the N gene, the S gene of the SARS-CoV-2 genome. And among these assays, RT-PCR assays targeting the RDRP gene has the highest analytical sensitivity. So that's why a majority of the kits targets either the RDRP or the ORF1AB. Now, depending on what your reason is for requesting for diagnostic tests for coronavirus, you have specific tools that you can actually use, maximizing. If you can use it for screening asymptomatic contacts and then monitoring them for the development of disease, to measure the distance to pre-incubation to incubation period to the onset of symptoms, or you want it to confirm an active symptomatic individual, whether they have the disease or not, or whether a decision to de-isolate patient when they are no longer testing positive, or you want to monitor the types of shedding. So it depends on your resources, depends on what your endpoint in terms of requesting for the test, but more often than not, it's really more on diagnosing active case and symptom-based detection, which still remains to be the most commonly used approach in using the diagnostic tools for COVID-19. Now, the timing of diagnostic testing in relation to the infection is critical in the utility and accuracy of COVID-19 diagnostics. If you understand the viral dynamics of transmission, you may still be able to use some of the existing technology depending on your purpose. If you are measuring active disease, or you are just measuring the number of exposed individuals in the community, then you can have a gamut of tests that are actually available already for you to utilize. Now, I've grouped the COVID-19 diagnostics into three major areas. The molecular-based testing, I grouped them to both nucleic acid and the antigen testing, and then serological testing, which is the antibody tests, and then the non-invasive procedures such as computed tomography in combination with artificial intelligence for image recognition software platforms. Now, for serological testing, there are three principal platforms that were actually studied in the systematic review published in the British Medical Journal, looking at enzyme-linked immunosorbent assay, lateral flow immunoassay, and chemiluminescent immunoassay. Now, the enzyme-linked immunosorbent assay, based on articles that actually passed the criteria for after their critical appraisal, had a pooled estimate sensitivity that is actually dependent at the time at which the sample were actually collected. If you test patients in the first week of illness, the pooled sensitivity is 26.7 for the IgM and 23.7% for the IgG. And as described in the curve previously, you will see a continuous increase in the sensitivity as the day passes. So you see its maximum utilization in the third week of illness, which has increased to 78.4 and 82.1, which is not a common, which is not unique to COVID-19. We see this in a lot of immunoassay serologically in some other infectious disease because of the dynamics of the immunological response and development once a person is exposed to the antigen. It's a pooled sensitivity based on among the infected population is about 84.3%, which is higher on the third week. And for non-infected, pooled specificity of about 97.4. Now, the lateral flow immunoassay similarly had the same behavior with the enzyme-linked immunosorbent assay. Its pooled sensitivity is lowest in the first week and continued to increase on the third week of illness for both IgM and IgG detection. And based on its pooled sensitivity among infected individuals, it is 66%. And the non-infected individual has a 96.6% specificity. The third serological tests is actually one of the much better versions from the first two. It's called the chemiluminescent immunoassay. Using fluorescence detections by chemical reactions, it allows technology to pick up the IgM and IgG by fluorescence tagging. Now, in the first week of illness, it had a pooled sensitivity of about 50% for IgM and IgG, and continued to increase on the second and the third week, which increased about 90.6% for IgM and 98.9% for IgG. Again, for positive individuals, it had a pooled sensitivity of 97.8%, and a pooled specificity for negative individuals at 97.8% as well. Now, this is the summary of the estimates of the diagnostic sensitivity of the test in terms of positive predictive values, which is actually dependent on the percent prevalence of the disease in a population. The higher the prevalence, the higher will be the positive predictive values, the lower it becomes low. But in all analysis, the pooled sensitivity has the lowest for the lateral flow immunoassays. But in this particular systematic review, take note that for each information generated here as a whole, the quality of the papers that has actually been studied are very heterogeneous with some high risk of bias in some of the designs. Sensitivity was higher at least 3 weeks after symptoms of onset compared within the first week of the disease. While this platform is not ideal or suitable for acute case detection, there may still be a role for serological testing if your intent is determining the burden of exposures in the community by a serological surveillance platform. So it still needs higher quality clinical studies assessing the diagnostic accuracy of serological tests for COVID-19. Currently, available evidence do not support the continued use of existing point-of-care serological testing for the acute detection of disease. The second is the molecular-based diagnostics. I'm sorry about the very busy slide, but this is a snapshot of what happens to the sample until you get to the result. Take note that for all molecular-based assay in this drawing, it starts with the sample collection. For those who are actually running laboratories, this is something that is very crucial, no? Because it's molecular-based tools is garbage in, garbage out. If the sample collection is not properly done, there is a tendency that you will not get the best sample, and you might be able to provide an erroneously low viral burden in the samples you are collecting. So the sample collection is very crucial as it affects the behavior of diagnostic sensitivity of molecular-based tools. So in this case, it's grouped into one, could be either an antigen-based detection assay, wherein the detection antibody has a label and it creates a colorimetric reporting once it attaches to the viral antigen captured by the antibody on the nitrocellulose membrane. The other one is by next generation sequencing, wherein you prepare a you do library preps of the sample by amplifying the DNA fragments, adding an adapter to be able to discriminate all of those samples in a pool. And a sequencing technology is done using either the Illumina next generation sequencing or a nanopore-based sequencing technology. The most commonly used globally is the PCR amplification, wherein you're actually using molecular probes with fluorophores that lights up every time there is polymerization of the gene targets that and then creates this curve down below, which is what you call a threshold value here. And then another one is the isothermal lamp amplification. You can either do colorimetric, or you can also do fluorescence detection using single temperature. You are actually able to amplify the gene targets, and using the lamp method wherein there is a 4 to 6 unique molecular probes that allows it to amplify, creating a loop mechanism, making it visible to the naked eye as the fluorochrome intercalates to every double-stranded develop. The more high-tech versions are using gene editing technique. In this case, you have the SARS-CoV detector and some others, which you can combine with a lateral flow reporter or other forms of chemical reporting system. So the entire global community who are developing diagnostic tests have utilized in part or in whole or even combined some of these platforms that I have just described. So for nucleic acid-based tests, the use of nucleic acid-based amplification products utilizes 3 usual platforms. The one that we're all familiar is the RT-PCR test. The other one is a digital droplet PCR, wherein a small amount of viral samples can actually be detected in a very small volume using digital droplet PCR, but the technology requirement to set up is a little bit pricey, and the reagents you need to create those droplets are also pricey. So, but it's really nice to know that it's actually more sensitive than the RT. The other one is for resource limited setting. The RT lamp can actually also be used. The reverse transcription loop mediated amplification technology, or the RT lamp under the nucleic acid based testing. In terms of the antigen test, you are actually directly detecting the viral antigens that are specific to the antibody, followed by either optical, magnetic, or electrochemical visualization of the interaction. And the targets for this are the nucleocapsid protein, the spike envelopes, and the membrane proteins as the viral antigens, for which the antibody that is placed either on a nitrocellulose membrane or any capture device. And then you add a detection antibody with the label that can fluoresce or change color if a viral antigen is actually complex. So this is the antigen detection. So comparing both molecular tools in terms of its intended use, it's really intended for acute infection diagnosis or active case findings. The analyte detected is a viral RNA for the RT-PCR and the viral antigens for the antigen test. And there are several types of sputum, swab, sputum, saliva, bronchoalveolar lavage for RT-PCR, and sometimes a swab test for the antigen test. The sensitivity, of course, is much higher for RT-PCR as opposed to the antigen test because of the limitations related to the antigen platforms. No? And test complexity is more complex in terms of the RT-PCR test, depending if you're using a cartridge-based single-use as opposed to 3 steps-based RT-PCR platform, so it varies. Well, for the antigen test, it's relatively easy because most of the designs are actually POC platforms. So there are some closed system RT-PCR detection kits for point of care technology, but majority of them really needs a laboratory. But for the antigen test, it's really designed to become a point of care. And the turnaround time, depending on if it's droplet PCR, you can detect the results in 15 minutes. If it's real-time PCR, depending on the burden of processing per day, it can range within 12 hours to 48 hours. But for the antigen test, it's about 15 minutes. The cost vary from centers, whether it's subsidized, fully subsidized, partially subsidized, or out of pocket. So the analytical performance of the nucleic acid based on study by Dean et al. Under the Cochrane database of systematic reviews, based on 13 studies, is 95.2%, with confidence interval of 86 to 98, and an average specificity of 98.9%, with a very narrow confidence interval at 97 to 99.5%, which is the usual behavior of a nucleic acid testing. This is based on 13 studies with an N number of 2,255. In terms of the antigen test, based on 8 studies, the average sensitivity is 56.2%, with a wider confidence interval of 29.5 to 79.8. Looking at the nuances of the average sensitivity, you can tell that the antigen tests are affected based on the quality of the manufactured product, and the antigen targets based on the one they are using and the type of capture that you have put in. So there is a range of sensitivities, which means if you are adapting an antigen test, it's very important to identify which type of product you're actually getting that has the better sensitivity studies, because once you average them, they get pulled down. And but I'm not gonna recommend any product here in this presentation. So nucleic acid-based platforms such as RT-PCR remain to be the gold standard for COVID-19 testing, and that improved primer and probe design and control over pre-analytical variables is expected to improve its clinical performance. Although antigen detection tests are limited by low accuracy, they remain as promising cost-effective platforms for population-wide mass screening, depending on which antigen test you're using as a point-of-care alternative to the more costly RT-PCR platform. Now, going to the other side of non-molecular based diagnosis, there are several studies that have utilized the use of computed tomography to describe the presence of specific structural abnormalities related to coronavirus. This is an example of a coronavirus cut section choosing ground glass opacity and some form of consolidation or organizing pneumonia with reverse halo sign. But in infectious disease, we see them also in other types of pneumonia, which make it very difficult to make a diagnosis just looking at the CT. But what it could actually help you if there is a strong epidemiological link to the picture, plus a strong clinical suspicion to the picture, then the likelihood of the diagnosis may be higher when you combine these 3 information. It has a pooled sensitivity based on 16 studies at 92%, but the specificity, as I mentioned, because other diseases can have the same finding, 25 to 33%. Due to the low contrast of the infection regions in CT images and large variations in both shapes and positions of the lesions in different patients, so the delineation of infection region in chest CT scan has always been a very challenging effort by our radiologists. So they added a composite of a three-dimensional reassignments and reassessment of the structure using the help of computer-based artificial intelligence programming. So instead of getting a two-dimensional photo, you get a three-dimensional hybrid of the disease that can provide you a much better assessments of the tomography of the disease, whether you have COVID-19 or not in this disease. So basically, it had improved. It has improved sensitivity as a single test, just looking at the image with AI, and a specificity of 79 to 99%. But it could increase if you add both epidemiological tool and the clinical profile of the patients. How can this be used in clinical? If there is a problem in turnaround time and there is an overloading number of cases at the emergency room, the utilization for may not be the best one, but can be the ones that can help triage patient is the utilizations of computed tomography and image recognition software using artificial intelligence plus clinical tools and epidemiological link to the patient can be used, but it really requires a lot of clinical acumen as well. So it's easier said than done. But again, if you're looking for alternatives in the absence of what is the best tool, then this can be one of the options for the diagnosis. So it is not advised to use chest CT as a sole diagnostic approach for COVID-19 infection. It is suggested that the chest CT may be necessary in patients with clinical symptoms, but had negative RT-PCR result, and still has a very strong suspicion. And of course, repeated CT scan may increase the risk of radiation exposure that may outweigh the sensitivity of chest CT in asymptomatic patients. Now, sequencing technologies has already been utilized for emerging infectious disease. In fact, it is through sequencing technology that the novel coronavirus was actually identified as novel. And recently, there were an FDA approval for authorization of the use of next generation sequencing for the diagnosis of COVID-19. This one is called the Illumina COVID-Seq, which is an amplicon-based next generation sequencing platform, which is authorized by the US FDA, which can produce 3,702 results in 12 hours with a positive agreement of 98% and a negative agreement of 97.4%. The word agreement here is used because when you are comparing two platforms, which is not really gold standard like the viral culture, it's concordance agreement rather than sensus specie. It's actually the one that is accepted in clinical laboratories. Now in this case, RNA extraction, and then you synthesize and amplify the cDNA, and then you tag them, you clean it up for background noise, you pull the libraries of all of these patients and run the sequencing platform, and you can have the results. The problem here is the cost, and the cost. So it's still not economically viable unless really there is partial subsidy in this case. The other NGS test that is available is the Helix COVID-19 that uses the Illumina NovaSeq 6000 sequencing system. Take note that each of these equipment I'm actually mentioning, we have it here in the Philippines. And so this is also an amplicon-based next generation sequencing platform authorized by the US FDA for sample pooling that can actually discriminate positive and negative results in such high throughput fashion. The LAMP4 nanopore sequencing is a combined multiplexed barcoded loop-mediated isothermal amplification, rapid barcoded library prep, and real-time nanopore sequencing that is CE marked for in vitro diagnosis CE mark is the code for European and some parts of Asia as a marking for clinical use. Others use, in the US, it's a US FDA counterpart. It can process up to 9,000 samples for 24 hours with a sensitivity of 90.1 and 99.6 specific. Unfortunately, the equipment and all of these things, the cost of the technology is very limiting. That doesn't allow for a massive testing for the entire country. The other one is the Ion AmpliSeq SARS-CoV-2 research panel, which is an amplicon-based, again, next-generation sequencing platform, still awaiting approval by the US FDA, but can actually be used for epidemiological purposes with a rapid turnaround time and high success rate, and can pick up as low as 20 copies of the targets in a sample. So the analytical sensitivity in pathogen detection is, however, limited with next-generation sequencing because of other microbial nucleic acid from resident microbiota that often result in a high background. In fact, over 99% of the reads are often from the human host, so that the depth of the sequencing must be high so that you can actually fish out the viral targets in a haystack of human genes. No? And large-scale population screening is limited by the high costs to perform next-generation sequencing. So there are several unmet needs in COVID-19 diagnosis. The scaling up access to diagnostic testing need more relaxed and streamlined regulatory requirements and procedures without forgetting the biosafety component of such procedures. Other alternatives to usual specimen types, collection device, transport media, and the need for clinical validation of the utility of other non-respiratory specimen may be needed as well in order for a more efficient but highly sensitive technology for the diagnosis. There's a need for short and medium-term pipelines for de-isolation decisions, need for more accurate screening of asymptomatics, and determination of the live viral shedding. We really need a lot of studies on this particular area. The standard quality assurance protocols need for wide-scale independent evaluation of emerging tests that's sometimes faster than our protocols being developed. And of course, the most crucial part is access. In low resource setting, the need for long-term scaling up of manufacturing, an efficient supply chain, and sustainability for in-country developments is something that we need to seriously think about, because during this pandemic, most of us were cut off from the supply chain because of the global problem, and we are on the waiting list all the time for technologies to arrive in the Philippines. So there are other emerging new diagnostic technologies from Foundation for Innovative New Diagnostics for point of care, manual NAT, nucleic acid test, RDTs, and automated immunoassays listed. And most of them are actually being evaluated all over. Now the more advanced versions of technology is the genome editing platforms. Technology combined with supporting detection technologies already available. This one is the Crispr Diagnostics that uses in the nucleus targeted CRISPR transporter, or using the CRISPR-Cas9 based assay for the detection of the SARS-CoV from extracted patient sample. It can perform simultaneous reverse transcription and isothermal amplification using loop-mediated RT-LAMP for RNA, followed by Cas12 detection of predefined coronavirus sequences, after which cleavage of a reporter molecule confirms detection of the virus. So there are several CRISPR platforms, the Sherlock, that is also available. And then you can also use the lab-on-chip, the proteome microarray that identifies proteomes of infected sera compared with controlled sera and determining their epitope level resolution, and to identify whether the patient has the COVID viral proteome or not. And it's called the per-chip SARS-CoV-2. Others uses biosensors to pick up the SARS-CoV in clinical samples, depending on which biological sensors you are actually using. And of course, there are other initiatives for the diagnosis that are available out there with some fancy names and companies using cartridge-based, smartphone-based, antigen test with ultra-sensitive single molecule immunoassay tech, and the like. No? So a number of diagnostic platforms currently exist. Deciding on what platform to use depends on the availability, diagnostic accuracy, time of infection, and purpose. Novel diagnostic techniques are continuously being introduced. Adoption of these technologies in the future will depend on regulatory policies, which are currently inadequate. So to navigate the maze of COVID-19 diagnostics, a multi-dimensional point of view should be adopted by test developers, policymakers, and clinicians. And these views should include the technicality of the test, the population to be tested, the intention for testing, and the regulatory monitoring of the test. Together, we can fight COVID-19. Thank you very much.
Thank you very much, Dr. Destura. May I clarify with the organizing committee first? Do we have a few minutes for Q&A? Dr. Carl, can I kindly verify that first? I have a lot of questions in the box.
Hello?
Yes.
Yes, ma'am.
Clarify, yeah. Can I just please clarify?
Go ahead, ma'am.
I'll go ahead. Dr. Raul, game?
Game.
Yes. The first question here, how can you explain the findings of persistent positive RT-PCR in an asymptomatic individual? Does it follow that if you had a long time to become negative, it means you really have a high viral load? If yes, how come you are asymptomatic?
Thank you. So it's just like any other virus that you're actually colonized in some form. Some individuals have the ability to harbor the virus but doesn't develop the disease. There are several postulates on that, meaning some of them do not have the essential targets for attachments, and some of them have their natural non-specific immune response to actually control the infection from actually creating the disease. No, there are a lot of postulates. That's why it's a big question mark from my end. If you look at viral shedding, there is no difference in the persistent viral shedding between symptomatics and asymptomatic. When you say persistent positive, the longest they've seen is up on the 4th to 5th week of from the time they actually had the test done. No? And does it mean continuous infectivity? The answer is I have no idea. But based on asymptomatic individuals in either long-term care facilities, taking care of them, there is an infection rate of 17%, as opposed to a Korean study that the infection rate is 56% to 60%. So clearly, there still need a stronger modeling on how to explain the behavior of persistent RT-PCR. The other thing is when you do viral shedding, when you are clearing out the virus, these are fragments of the virus that still retains the protein surfaces or the targets that are being used by RT-PCR. Hence, they would still test positive, which and they may no longer be infectious. So after a very long answer, the answer really is I really don't know. Very difficult to say.
Yes.
Yeah.
Here's a quick question lang po. What happened, or is there any update on the validation of pooled testing proposed by the Philippine Society of Pathologists?
I don't have direct knowledge on the pooled testing, but my take on that is the value of pooled testing depending on the prevalence of the disease in the community. If the prevalence of the disease is higher and you do pooled testing, chances are everything will be positive. And then when you pool them and one sample tests positive, you will spread it out and test it individually. Now imagine the two-step procedures that you will be doing. So if the prevalence of disease is low, let's say less than 2% or less than that, then the role of the pool testing will actually work. No? And but if the prevalence is high, then it might no longer be cost effective. Second, there is a timing for collecting. There's a strategy for collecting for pool testing. They need to be in a community-based or in a location-based. It cannot be sporadically collected all over the place and then you pool them, because the decision to isolate depends on where they are. So if the design is properly thought of and the prevalence of the disease is low, maybe among, let's say, asymptomatic groups, maybe there may be a role, but still needs some studies to be done. So I wish I'm not privy with what's going on with that proposal.
Yes. Actually, we are really running over time, Dr. Destura. We have a lot of questions in the box. Is it safe to say that you can stay a few minutes to just type in the answers to all these?
Okay, sure,
Yes.
sure.
Just one more question from Dr. Alora about a positive test becoming negative, then positive, then negative during a 2-month period in an asymptomatic patient.
Yeah, I've seen that a lot of times, no? And sometimes the test is being done by different laboratories looking at different targets. So sometimes the cycling, the test are you're testing for this gene, it tests positive, and then when you look for the other gene types in another laboratory using a different kit, it's negative, and then you go back and forth. But let's say you are using same test all the time, no? Low virological burden in the area can actually affect the likelihood of picking up the virus or not. So again, if the collection is wrong, if the pre-analytic processing is wrong, then chances are it can test positive now, it can test negative later. But whether if you test positive now and then 2 months later you test positive again, I don't know if it's persistence or another person you got exposed to another individual. So again, we need more studies to be able to answer that question.
Okay, Doc. So, Doc, the questions in the box, kindly please do type in, and then also with the organizing committee can take note of that. So all your questions are noted and will be answered later. So
Thank
we would
you,
like to
Mitzi.
thank you so much, Dr. Destura. Very, very informative topic. At this point, may we flash the certificate of appreciation for Dr. Destura so that we can continue. There you got it. I won't read it anymore, but our most sincere thanks and congratulations again, Dr. Destura. Dr. Lagamayo, thank you so much on behalf of both of us. I'm really under time limit here. Thank you so much. The next session will focus on from diagnostics to move on to therapeutics. So what are the treatment options for COVID-19? Please wait for the session chair, who will be Dr. Myrna Mendoza. Thank you so much to everyone.
Good evening, everybody. From diagnostics, we go now to therapeutics. The next topic will be on therapeutic options for COVID-19. And this will be tackled by none other than our president, Dr. Marissa Alejandria. And I'm proud, and it's my pleasure to present her at this time. Besides being the president of the society, Dr. Alejandria is professor and associate dean for research at the UP College of Medicine. She's also director of the Institute of Clinical Epidemiology, National Institutes of Health, UP Manila. At the same time, she is a clinical professor, Division of Infectious Diseases at the Department of Medicine, UP PG, at the section of Infectious Diseases, Department of Medicine, the Medical City. Ladies and gentlemen, Dr. Marissa Alejandria.
Good evening, everyone, and thank you, Dr. Myrna. It's a privilege to be introduced by my mentor. Thank you, ma'am, for the kind introduction. So I'll share my screen. So I'll be walking you through the evidence on the available treatment options for COVID-19. It's a challenge preparing this lecture because, as you know, the avalanche of literature over the past months has really been very, very fast. So the landscape in the treatment of COVID-19 is really evolving, and new trials are coming out. Sorry. Yeah. So new trials come out, like for instance, in the WHO database, more than 120,000 studies are there, and that's like about one study every 2 minutes that you need to review. Okay. So for this evening, the objective given to me is to discuss the various therapeutic options, including antivirals, immunomodulators, and other therapies under investigation for COVID-19, and to describe the contribution of the Philippines in the Solidarity Trial. These are my disclosures. I'm currently the National Coordinator for the Solidarity Trial and a member also of the Executive Group of the Solidarity Trial and International Steering Committee. So if you listened last night, so Professor Yi Xin, our speaker on the inflammatory response to COVID-19, ended with this slide, and now I will start with this slide and review you on the pathogenesis and therapeutic targets for COVID-19. Okay, so as we all know, so the spectrum of COVID-19 starts with the mild constitutional symptoms during the first week, or that's the stage 1, the viral response phase. And then it moves towards the host inflammatory phase. So depending on the host response, so you either recover or you move into the pulmonary phase. And the pulmonary phase can be divided into the phase wherein there is no hypoxia, and then if the immune response further goes reacting, then you move into the post-inflammatory response wherein you now develop hypoxia, or you may proceed to ARDS, now the critical phase. Okay. So from mild, moderate, mild is, if we review, just constitutional symptoms, no hypoxia, no pneumonia. For the pulmonary phase, that's you have the phase wherein you only have low flow or supplemental oxygen with a oxygen saturation of less than 93 or less than 90, depending on the cutoffs that are being used by different institutions, and then move to the pulmonary phase with requiring high flow oxygen or mechanical ventilation, wherein you now experience the cytokine storm. Okay. So for each of this phase, there are targets, no? Potential therapeutic targets, and it can be divided into 3 major targets. The viral entry is one potential target. So we have groups of drugs that which aim to prevent viral entry, like chloroquine, Camostat, Arbidol. Okay. And then the other group is the one that inhibits viral transcription, viral translation, and viral replication, such as your lopinavir, ritonavir, remdesivir, and favipiravir. And the other big group is the one that targets the inflammatory phase. No? So we have here our dexamethasone, which inhibits inflammatory cytokines, neutrophil infiltration, and then the monoclonals or immunomodulators such as the tocilizumab, which targets specific cytokines or interleukins, and then the monoclonal antibodies and convalescent plasma, which is a form of passive immunotherapy, wherein they produce neutralizing antibody for convalescent plasma, and we have monoclonal antibodies that neutralizes the virus and preventing viral entry as well. Okay. So that's a review on the therapeutic targets, and we can see here that we have some of the drugs that are targeting these 3 main areas. So what are the options for mild COVID-19? So to date, supportive care is still our mainstay for the management of mild COVID-19, and this includes antipyretics for fever, oral fluids for hydration, and we have to isolate the patient at home if they have the required conditions to enable isolation, or in temporary treatment and monitoring facilities. This is a strong recommendation, and we also strongly do not recommend the use of routine empiric antibiotics and routine anti-influenza drugs among patients who have mild COVID-19 disease. And this is based on systematic reviews which have shown that bacterial co-infection is low on presentation. So on admission, it's about 3.5%. It's only when patients move into the severe or critical stage that the bacterial infection prevalence goes up to 8.1% or even up to 14%. Another systematic review also showed similar percentages. So overall, 7% of hospitalized COVID-19 patients had bacterial co-infection, and a higher rate among those in the ICU, while viral co-infection was only 3%. So hence, we should preserve our antimicrobials for severe and critical COVID-19, and should be used empirically only for severely ill COVID-19 patients. And if we do start antimicrobials, we should regularly assess their necessity and monitor for possible side effects. And once we have cultures, either de-escalate or switch to oral as soon as possible. So again, no antimicrobials for patients with mild and moderate COVID-19. And we have to here to guide us on which patients we need to give antimicrobials and what antimicrobials we should give. We have to be diagnostic stewards as well. So use workup, so as discussed earlier, so you can use chest CT scan biomarkers to guide us if we are dealing with just pure COVID-19 or a co-infection. And if we are to start antibiotics, we have to do our get cultures. Okay. Now, how about the antiviral interventions? What's the evidence now for hydroxychloroquine with or without azithromycin, lopinavir, ritonavir, and favipiravir? Okay. So these are just some of the, these are the key randomized controlled trials on hydroxychloroquine with or without azithromycin. And you can see here that all of them showed no difference in terms of mortality for the big recovery trial, no difference in conversion to a negative SARS-CoV-2 PCR, No difference in incidence of COVID-19 for those who have used the trial on post-exposure prophylaxis. No difference in clinical status, no, for the clinical trial on hydroxychloroquine with azithromycin or azithromycin alone. So I'd just like to highlight this big trial on this multi-center adaptive trial in UK, the recovery trial, which gave us a conclusive evidence that hydroxychloroquine is not beneficial for hospitalized patients. No? So it did not show a significant difference in mortality. And likewise, the randomized controlled trials on lopinavir-ritonavir did not show any benefit in terms of, again, mortality for the big trial of recovery and then in terms of clinical improvement. Again, to highlight the biggest trial on lopinavir-ritonavir, which showed no significant difference in the primary endpoint of mortality among hospitalized patients. And with the results of the recovery trial during that's about the third or fourth month of the pandemic, the Solidarity trial did an independent interim analysis as a result of the recovery trial, which showed no significant difference, no, in terms of mortality. And likewise, the WHO Solidarity trial showed that hydroxychloroquine and lopinavir-ritonavir did not benefit patients in terms of reduction in hospital mortality. Hence, the Solidarity trial stopped the hydroxychloroquine and lopinavir-ritonavir arm. So this RCTs, no, especially the two big RCTs, Solidarity and Recovery, served as the basis, no, for the strong recommendation that chloroquine or hydroxychloroquine as monotherapy in combination with macrolide or another antiviral agent among hospitalized patients with COVID pneumonia is not recommended. It is also likewise not recommended for outpatients with early or mild COVID-19 disease, except in the context of a clinical trial. The same recommendation is made for lopinavir-ritonavir. It's not recommended as a monotherapy or in combination with hydroxychloroquine or other immunomodulators. And IDSA has the same recommendations, no? Again, these are strong recommendations on not to use hydroxychloroquine for mild to moderate COVID-19 or even among hospitalized patients. Okay, now let's move to another set of drugs, no, that targets now the viral replication. That's favipiravir and remdesivir. So for favipiravir, so it inhibits the RNA polymerase, halting viral replication. It's a drug that has been approved for treatment for influenza in Japan in 2014. There are 9 ongoing clinical trials on favipiravir. However, all of these trials are still on preprint, and one is a press release. And for the early trial, no, this is the earliest trial in around March or April, which showed, this one is comparing favipiravir against umifenobir, also a drug that whose mechanism of action is preventing viral entry. This drug, no, favipiravir, showed shorter time for fever reduction, cough relief, and prevention of new onset dyspnea, but no significant differences were observed in the severe and moderate arms. No. Okay. And then just this month, these are the 2 and other additional trials, no, but again, still preprint, and these preprint trial was done in Russia with 190 patients, okay? And the other one is India with 150 patients, and it's accepted for publication in IJID. So we have these small trials, open label, The one in Russia, however, is compared again against an active control, and the one done in India was compared against best supportive care. These two trials showed improvement in time to resolution of symptoms, or clinical median time to clinical improvement was 6 days in the favipiravir group and 10 days in the control arm, which is umifenavir plus interferon. So there was a difference of 4 days. And while the trial from India showed a difference of 3 days for time to clinical cure for favipiravir and 5 days for the control arm. So a difference of 2 days. So, and then the Japan trial is still just a press release. We have not seen the publication, which in this Japan trial is also a small trial, which again claimed to have a shorter time to resolution of symptoms among those who receive favipiravir. So with these 4 small trials and the outcomes being mechanistic just on time to improvement in clinical symptoms, we await still another multi-center clinical trial that's currently ongoing in the country, which also is enrolling patients with non-severe pneumonia. So because of this insufficient evidence, based on small trials, we do not recommend routine use of favipiravir in the treatment of COVID-19, except in the context of a clinical trial or for compassionate use, as it is just not available commercially, especially in our country. It's for compassionate use among patients with mild to moderate COVID-19 disease given at this dose. Okay, now let's move to the more controversial drug now, the remdesivir, the most popular and most controversial drug as well. So these are the 4 trials that we will review, and which is the basis for the recommendations, the conflicting recommendations between IDSA, NIH, and WHO. So allow me to present to you the WHO Solidarity Trial results on the repurposed antiviral drugs for COVID-19. So this has been submitted to the New England Journal and accepted for publication and will come out sometime soon this week. So we've also submitted it as a preprint in the medRxiv database. So for the Solidarity Trial, we were able to enroll 11,266 adult patients from 30 countries and 405 hospitals. The interim analysis is as of October 4, so it includes patients with outcomes as of October 4, and these are the numbers of patients randomized to each arm. So 2,750 for remdesivir, 954 for hydroxychloroquine, 1,411 for lopinavir, ritonavir, and 2,000 for active interferon. 651 were combined with lopinavir, ritonavir, and 1,400 plus were combined, was given solely as interferon. So this happened because we had to stop lopinavir by the 3rd month of the trial. So by the 4th month, we were just giving interferon alone. Okay, so in terms of follow-up, so we have about only 0.6% of the study population at this point where did not have outcomes, because this group, these patients were the ones who did not consent for their outcomes to be reported after they have been withdrawn from the trial, either because of deterioration of clinical condition or they wanted to be shifted to an active arm. Okay. Now, some highlights on the demographic data. So majority of the patients that were enrolled in the trial were less than 70 years old, so 80%. And then majority were on oxygen upon admission, entry into the study. Majority had bilateral infiltrates. 62% were men, and the most common comorbidities were heart disease and diabetes mellitus. In terms of geographic location, 61% were from Asia and Africa, 17% from Latin America, and 22% from Europe or Canada. So we've contributed around 1,000 plus patients, no, in this analysis. Okay. And in terms of days in the hospital, 38% were already on the second day onwards when they were recruited into the trial. Now let's go to the results. Okay. So in terms of the 28-day mortality or probability of death, we see here in this Kaplan-Meier curve, survival curve, over 28 days that there was no difference between remdesivir and the control arm. Likewise, no difference in the hydroxychloroquine and the corresponding control. Same thing is seen with lopinavir, ritonavir, and interferon. So all four drugs did not show significant difference in reducing hospital mortality. Now let's look closely at remdesivir. When stratified by age and oxygen support, here we also did not see any difference. However, if we look closely at the ones, if we stratify in terms of ventilatory support at entry, those who were not ventilated upon entry into the study seem to have a trend towards benefit here with 0.86, but although the confidence interval still crosses 1. This is just a graph to again show that there is no significant difference when we stratified, no, by age and by ventilation. But as you stated, there was a tendency towards benefit among those who were not yet ventilated. And for the secondary outcomes, we also look at whether those who were not ventilated at entry, did they progress, and is there a difference between the control arm and the treatment arm? And here are the absolute numbers, and we see no significant difference, no, in terms of the number of patients who were already on ventilator at entry, who were not already on ventilator at entry. There was also no reduction in hospital stay with any of the study drugs. No. The percentage of patients still in hospital on day 7 were similar with the concurrent controls for the 3 drugs. And as this graphs here clearly show, no, that there is no difference, and none of the drugs substantially reduced recovery time. Now we did a meta-analysis to compare, no, the results of the Solidarity trial with the NIH trial and the other 2 trials, the trial done in China, which was stopped prematurely because they do not have cases anymore, and then the other trial published in JAMA, which was recruiting moderate cases, no, moderate COVID-19. So if we look at the this meta-analysis, which stratified the trials in terms of oxygen use. So for the Solidarity trial, when stratified by ventilation, so again, no difference. But as I mentioned earlier, those on low flow or no oxygen had a tendency towards benefit. The same thing is seen with the ACT trial. No? So in terms of mortality, you see here a benefit among those who are receiving low flow oxygen, but no benefit if they are already on high flow and invasive ventilation. And for the smaller trials, we did not see any difference as well. So if we combine the trials, the patients, the subgroup of patients who are on a lower risk, meaning they are on no oxygen or low flow oxygen, we again see a trend towards benefit, but no benefit for those who are already on high flow oxygen or on invasive mechanical ventilation. Okay. So overall, the 4 randomized trials of remdesivir versus standard of care or without it have been reported. Okay. The Solidarity provides 3/4 of the evidence on mortality. So in terms of the weight that it gives into the pooled estimate, it contributed 604 deaths compared to just 136 deaths in about 1,000 randomized for the ACT trial. And the other 2 smaller trials had, of course, smaller contributions. And while the ACT trial found that remdesivir moderately reduced time to recovery, the Solidarity trial had no effect on numbers progressing to ventilation or on time to discharge. So combining the data from all 4 trials, we see that there is no significant difference in terms of mortality. And this slightly favorable remdesivir result could have been due to chance if remdesivir does nothing and the real death ratio is 1 in all the trials. There could also be a small benefit in lower risk patients, but not a big one. So here we see that more evidence is needed for precision, but better drugs are needed even more. So remdesivir cannot do it alone. So just to review, the ACT trial, which enrolled more than 1,000 patients, and their primary endpoint is time to recovery. They showed a reduction in significant reduction in recovery time in the remdesivir arm. And when they stratified it by oxygen support, the benefit was seen among those who are on low flow oxygen, but not on high flow and mechanical ventilation. Okay, so again, this is the forest plot, which shows those who are receiving low flow oxygen are the ones that had the fastest time to recovery. And then the other trial, the smaller trial on moderate COVID-19, which compared 5 days, 10 days, and standard of care. Okay. Again, this also showed that patients who are receiving 5-day remdesivir were more likely to have clinical improvement compared to standard of care. And no difference was seen between 10-day remdesivir and standard of care. And this is the small trial from China, which showed no difference in time to clinical improvement and duration of mechanical ventilation, but it did not reach its required sample size. So again, we go back to the 4 trials and we see that the biggest trial is the Solidarity trial, which contributes to most of the evidence, and it's across 405 hospital sites and 30 countries. And if you look at the percentage of patients in terms of severity, the ACT trial had a greater percentage of patients who are in the severe or in the critical stage. No? Okay. While the Wang trial only included severe patients, while the Spinner trial, which is published in JAMA, only covered non-severe or the moderate cases. So let's examine why WHO came up with a conditional recommendation against the use of remdesivir. So it suggests not to give remdesivir to hospitalized patients regardless of disease severity. And they did also a systematic review of the 4 trials, and they showed no difference in terms of mortality, mechanical ventilation, serious adverse events leading to discontinuation of the drug. There was also no difference in terms of time to clinical improvement, duration of hospitalization, and duration of ventilation when all the 4 trials were reviewed, as shown in this evidence table. So considering the low or very low certainty of evidence for all the outcomes, the guideline development group, I'd just like to emphasize here that the WHO guideline development group is independent from the WHO Solidarity Trial Secretariat. So we did not participate in the formulation of this guideline. So what the guideline development group is saying is, based on the evidence, it did not prove that remdesivir has no benefit. Rather, there is no evidence based on the currently available data that it does improve patient-important outcomes, particularly mortality, need for ventilation, and time to clinical improvement. Greater value is placed on being able to demonstrate evidence of efficacy, since this is a new drug, which was not established by the currently available data, since all the patient-important outcomes showed no difference. And the panel also placed a low value on small and uncertain benefits in the presence of the remaining possibility of important harms. While there was no difference in serious adverse events, pharmacovigilance still needs to be done. So you need to monitor the patients who receive remdesivir. With regards to the subgroups, we've seen that there is a tendency towards benefit among those who are non-severely ill or not receiving oxygen or just on low flow oxygen. The conclusion there is that remdesivir treatment possibly increased mortality in the critically ill and also possibly reduce mortality in the non-severely and severely ill. However, the panel thinks that the overall credibility of the subgroup effect is insufficient to make subgroup recommendations. There is no a priori hypothesized direction of the subgroup effect by the investigators. There is little or no previously existing supportive evidence for the subgroup finding, and the cut-off points were arbitrary to examine the subgroups of interest. And then the panel also considered contextual factors such as resources, feasibility, accessibility, and equity for countries and healthcare systems. So an opportunity cost was considered, knowing that remdesivir is an IV drug and it's not cheap, and the availability is also an issue. So these factors were considered, and if opportunity cost is to be considered, if there are issues on finances, then it would be better to invest the funds on dexamethasone or corticosteroids, which has been shown to conclusively reduce mortality among patients who are on oxygen or in the severe and critical stage. Hence, continued enrollment in clinical trials is encouraged. The IDSA recommendation came up also with a conditional recommendation, but they suggests against remdesivir for routine treatment of patients with oxygen saturation of more than 94% and no supplemental oxygen. And also strongly urge continued recruitment into the into clinical trials. So this is, I think, consistent, no, with the WHO recommendation for this part, and it is actually this part that where there is a disagreement, no, with here the guideline panel suggests remdesivir rather than no remdesivir for treatment of severe COVID-19 in patients with oxygen saturation less than 94%, including patients on supplemental oxygen, on mechanical ventilation, and on ECMO. In contrast to the WHO recommendation, which recommends against remdesivir regardless of severity. Likewise, IDSA encourage additional clinical trials to provide increased certainty about the potential for both benefit and harms of treatment with remdesivir, and to understand the benefit of treatment based on disease severity. So essentially, we are in a clinical equipoise as far as remdesivir is concerned. So we are in a debate whether to give or not to give remdesivir, and in a clinical equipoise setting. So how do we advise our patients? So we have to just clearly tell them the results, no, of the trials that they are conflicting. There is, with regards to mortality, they consistently show no benefit in terms of mortality, but in terms of clinical improvement, that is where there is debate. No, the trials are conflicting in terms of their results. So therefore, you may have to consider the clinical circumstance, the socioeconomic circumstance, the values and preference of your patient when deciding whether to give or not to give remdesivir, pending more trials that will definitely tell us whether there is really a benefit or not as far as reducing mortality is concerned among patients, among severe and critical and moderate COVID-19 patients. So what are the ongoing clinical trials in the country? So as we continue to enroll patients in the Solidarity trial for the remdesivir arm, and discussions are ongoing on what additional arm we are going to include into the trial. And we will know most likely next week or succeeding weeks, no, whether we are going to include monoclonal antibodies into the Solidarity trial. And I mentioned the favipiravir trial, which is headed by Dr. Nina Berba. And then we are also part of the anti-coronavirus therapy trial to prevent progression of COVID-19. This is also a multi-country trial funded by, this is through the initiative of Population Health Research Institute of Canada. This is an outpatient trial comparing aspirin versus control and colchicine versus control. So it's a run using a factorial design. And then locally, we have ongoing trials on melatonin, convalescent plasma, tocilizumab, and then the herbal drugs, so VCO, Lagundi, and Tawa-Tawa. Time. Okay, since in the interest of time, no, so just quickly again, just show you the what I mentioned earlier, that it's only corticosteroids that has been shown, no, conclusively to reduce mortality among critically ill patients, and as shown by this systematic review, no. So there are several trials, and the biggest one is that again of recovery, and this is the basis for the strong recommendation for the use of steroids for severe and critical patients, and it's not recommended for the non-severe COVID-19, those who are not on oxygen. Okay, so the same thing is for the recommendations are consistent, no, with NIH and IDSA. For tocilizumab, our favorite immunomodulator, We also have conflicting results on this. No? So IDSA actually suggests against the routine use of tocilizumab. So the COVACTA trial showed no significant difference in mortality, while the EMPACTA trial showed benefit. So there is still insufficient evidence, no, as far as tocilizumab is concerned. Now for convalescent plasma, likewise, the trials, no, the completed trials show no significant difference in terms of mortality. So the most recent publications, one that was done in India, also showed no reduction in progression to severe COVID-19 or all-cause mortality. So this one is for moderate COVID-19, not showing benefit as well. And this is another one on published in New England Journal for severe COVID-19. So again, we did not see a difference in mortality. So for convalescent plasma, there is still insufficient evidence as well. No? So we do not recommend routine use except in the context of clinical trials. Just to show you the contribution of the Philippines to the Solidarity trials, this is what we have recruited so far. As of yesterday, we have 1,238 patients into the trial. So 438 allocated to remdesivir. And as this graph reflects the prevalence of cases in the country. So we see a decrease in the number of cases in NCR, and we also see a decrease in recruitment. Okay, so, and among the public hospitals, we have recruited 661 patients, 577 among the private. So, and I'd just like to thank all the investigators who have committed their time in recruiting patients for the Solidarity trial. So we have 26 hospitals included in this Solidarity trial, 9 public, 5 provincial, and 17 private hospitals. So I thank everyone, the site investigators and their teams for the dedication in recruiting patients for this Solidarity trial. And I just like to end with some research gaps. So we've seen that still there is a need for better drugs. Remdesivir alone cannot do it. So thus far we just have dexamethasone or corticosteroids. So we need to look at combination regimens. As you've seen in the pathophysiology, we have a viral and inflammatory phase. So, and then the timing of administration might need to be studied further, which is we need to go back to our patients, go back to the cohorts. We might be able to generate some hypothesis so that will guide the direction in terms of the treatment arms or the combination regimens that need to be studied. And we also need studies on prognosis or prognostic markers to guide us on which patients will progress to the inflammatory phase. So we continue to work in solidarity in search for safe and effective treatments to save lives. Thank you for listening.
Okay, so thank you, Dr. Alejandria, for the lecture, for the very comprehensive lecture. So can we please flash the appreciation certificate for everyone to see? So I'm not going to read the certificate again, but this one would like to thank you, Dr. Alejandria, for giving us a very comprehensive and very thorough lecture about the treatment options for COVID-19. So actually, we do not have enough time, but if it is okay for Ma'am Isa to just type in the questions for the viewers, if okay, Paul.
Okay.
Okay, so I think okay for Dr. Alejandria to just probably stay and type in the questions in
Yeah.
the Q&A box now.
Yeah,
Thank you very much. So now we'd like to open the next session. So this one will be chaired by Dr. Berba. Thank you very much. Good evening.
Okay, so good evening and good morning, everyone. I'm Regina Berba, and I'm the chair for this session. It's Plenary 7, entitled Long Haul COVID-19, Close and Long Encounter with the SARS-CoV-2. I feel extremely honored to be here and part of this session and have the distinct privilege to introduce our next esteemed speaker. Dr. Paul Garner is Professor of Infectious Disease at the Liverpool School of Tropical Medicine, which is in the United Kingdom. We know him best as the coordinating editor of the Cochrane Infectious Diseases Group. And because of his team's work, the review portfolio has helped shape policies in the control of malaria, tuberculosis, and other neglected tropical diseases. Dr. Garner also runs the Center for Evidence Synthesis for Global Health, and he is director of the Research, Evidence, and Development Initiative, or the REDIT, and the person responsible for the WHO Collaborating Center for Evidence Synthesis for Infectious and Tropical Diseases. Now, in March 19, 2020, Dr. Paul Garner was diagnosed to have COVID-19. And in early May of 2020, he released his story. His personal account was one of the first published narratives that people at home could have a protracted illness in the BMJ. The Guardian report that followed had 5 million views within 24 hours. And in further 4 blogs over the next few months, he described the relentless course, the exhaustion, the mental fatigue that was very prominent. Dr. Garner has described the strategies he found that helped him recover. So to tell us firsthand about this phenomenon, please help me warmly welcome Dr. Paul Garner.
Thank you so much for that introduction, and I'm very pleased to be here and back with my friends and listening to Marissa. Actually, we've known each other for a long time, working on IVIG many years ago as a Cochrane review. And so it is pretty strange for me to be a person that works in systematic reviews and meta-analysis of thousands of people to be talking about myself in N equals 1. And also to be talking about myself. I don't normally talk about myself, but this did seem a particular point in time where it did seem relevant, where I had something to say. Can I just check the sound is okay for everybody, just so that I'm not proceeding and nobody can hear me?
Yes, Dr. Garner, we can hear you.
Okay, thank you. So, and so I'm very privileged to be here, back among friends. So thank you so much for this. And also, I also don't want to go on for too long about this. I'd much rather then open it up for some questions and discussion. So I should say I'll talk for 15, 20, 25 minutes maybe. At the very most. But you know, I'm in my 60s. I in March last year, or March this year, it feels an age ago, I must say. I was running a lot. I felt pretty. I was doing British military fitness. I'd suffered, you know, in the past. I'd had dengue and I'd had malaria and I'd had a ton of other things. And so COVID was on the scene, and I didn't really see it. I didn't at the time think it was going to be much of a threat to me. And suddenly I got COVID in March 19, and actually I couldn't influence what then happened next, really. And I felt COVID was, the UK government was pursuing a policy of herd immunity. We were preparing to lock down. I was going into work. I was checking my temperature every day. I didn't have a cough. I didn't have a temperature. I had no shortness of breath. But I was on a conference call with Nabarro in WHO, David Nabarro, and he said, this disease has a doubling time at the moment of 3 days. If anybody feels strange or unusual in any kind of way, they must self-isolate. And I started feeling, I felt a little bit strange. So I thought, well, maybe I better be careful. I rushed home and self-isolated. My staff were all furious with me. They thought I should, as a doctor, demand a test, but there were no tests available at the time, and so on. So it was a very difficult time. And the first few days, I didn't actually think I was particularly unwell until a sort of headache kicked in, and you would have all heard the various stories about this. And then suddenly, bang, it came out of nowhere. I almost collapsed. I deteriorated with very a severe I had some shortness of breath, a rapid pulse. I had a blinding headache and absolute fatigue. I couldn't lift my arms or do anything. And so the only thing I could do was go to bed. And I lay in bed and I thought this must be a myocarditis or something. I was Googling myocarditis as I sort of passed out. I mean, lying down, passing out is a pretty weird thing to happen. And I actually thought I was dying at the time. And a number of other people have noted this very severe exhaustion and systemic effects being related to this. Maybe my SATs were down, I just don't know. But a few hours later, I woke up and there I was. Then I started this illness that then went on for just, well, months actually. And as you will have come across with your own selves and with your own families and others, it just sort of, the disease is completely random. It goes away a bit and you feel a bit groggy, and then the next morning you wake up, you've got feel as though you've drunk a bottle of Sambuca and run a marathon, and you have ringing in your ears. You just feel completely wrecked, and then the next day you feel a bit better, and it seems to have this cyclical trend, and every day you think, oh, I feel better today, or I feel worse, or it's gone, it's going to go. And at the time, the British government was saying, and everybody was saying, this disease lasts for 14 days, maybe 3 weeks if you're unlucky. And so every day I was Googling these recommendations, and every day I was looking at my clock saying, well, I'm still not well. And at 6 weeks, I was in touch with Fiona Godley in the BMJ. I said, there's something weird going on here. You know, it's just going on forever. So I wrote the first personal view about it. And indeed, in those first 6 weeks, there was every day was something new. Like I'd get an acutely painful cough one day. I'd have tinnitus. I'd have buzzing in my arm, sort of this paresthesia, the tingling in your arms and in your legs. I was breathless one day. I woke up with palpitations in the middle of the night once. I woke up with fasciculation in the middle of the night. One morning I woke up and I had arthritis in my hands. I had weird sensations on my skin one day. I'd be acutely sweaty at night. I'd get up in the morning and I'd feel cold. I'd put 4 or 5 layers of pullovers on because I felt so cold. I mean, it was really bizarre. And the other thing is I'm a pretty resilient character, but God, it gets to your mood as well. So you must be careful with your patients about what's going on for them. You'd wake up in the morning with your head full of tears and pain and just crying. and then by the afternoon you'd be fine. And so the virus and the effects of the virus do these things to your mood. They are not just because you're in lockdown or just because you are ill. The virus affects your mood in very dramatic ways, and buzzing in your head and ringing and so on. And so that went on for months, and I just and the response to the first article was very interesting, because there was a flood of emails came in that people said, and because we were all nursed at home, I hadn't seen a medical practitioner at this point. I was entirely alone at home. And so was everybody else in the UK, because everybody was instructed not to go to hospital. And if they dialed 111, they'd just get a bit of advice and stayed at home. And so after this article came out, and the Guardian newspaper article came out, there was a slew of emails with people saying, thank God you said that. I've been going through this madness for months. I thought I had gone crazy. There were people that thought that they had developed a mental illness during this time because it was so strange. I had weird experiences of friends in Australia who were working in hospitals sending me photos on WhatsApp of a picture of me on the front page of the Guardian saying, I hope you'll get better. It was this sort of public face of being ill was extremely odd. But, you know, the point was that letting people know what was happening and that it was normal was I think, helpful to people. And then I started turning to some of the Facebook pages, and there was a lot of people describing similar things, or similar and different things. But there was a real sense that these people were having all of these symptoms dismissed by their families, and also at that time by doctors. So people would go to their doctor and say, I've got a very rapid heart. A friend who's a psychiatrist went to a casualty department and said, I've got a really rapid heart rate, which we now know is part of long COVID. She said, I've got a really rapid heart rate and I feel really dreadful. And the casualty officer just said, you're anxious. You're just anxious about the disease. This has nothing to do with COVID. And she said, of course I'm anxious, because I've got a very rapid heart rate to do with COVID. So people, you know, people obviously get anxious when they have these bizarre symptoms, but it doesn't mean that the symptoms are due to the anxiety. So I think it's also important that practitioners remember that. And I had some fascinating experiences with my occupational health doctor at work, who said, who interviewed me and then refused to say that I had COVID because I hadn't got a test. He said, this man has had some kind of severe illness, perhaps a virus. And I refused to sign off on it because it was so outrageous. So you were seeing a professor in evidence-based medicine talking to a doctor, saying how ill he was that was completely consistent with COVID, and them saying, oh, it couldn't be COVID, it has to be something else. I mean, actually reflecting back on it, you know, I didn't let you know, but I had anosmia very early on. And the reason I knew that was there was a couple of people living in the house at the time, doctors, and we were bleaching all the surfaces to keep everywhere clean. And I thought the bleach was inactive. I thought I couldn't smell it. So I thought, oh, I must have had it for too long. And I was bleaching the surface, and I thought, well, it's just, you know, it's just gone off, and so put my hands on the cloth and then took my hands off, and my skin was all sort of peeling off. It was horrible. But so there was that sort of 3 months of this intermittent illness that kept coming and going. And then it moved into a more chronic sort of feeling ill most of the time, with feeling unwell, having also acute episodes, dramatic mood swings I've talked about, this brain fog that came in. So this was when the sort of chronic fatigue set in, and the brain fog really is you can't think straight. I couldn't read long documents. I could read a short email, but a whole document, a whole email, I just said to people, I just can't see the words. I'd forget words. When I was trying to speak, I tried to talk to somebody about a review, and I couldn't remember the word for excluded. I said it's discluded, and I spoke to doctors about this, and they thought I was having a stroke, and that I should have an urgent MRI. I put this symptom on the Facebook page, and everybody said, "Oh, we've been having that symptom for ages." I told my husband to get those round white things out of the cupboard because she couldn't remember the word plates. Somebody told her husband that she was trying to say that she had an appointment, and it came out that she had an accountant, and he thought she was having an affair. I mean, it was extraordinary, this sort of muddling of words that was happening as part of this sort of post-viral whatever it was that was going on with us all. So at that time, I turned to websites to try and get some help. And of course, the NHS and other services are usually pretty terrible around dealing with post-viral syndromes and pretty terrible with dealing with people fatigue. They are, usually dismissed, largely dismissed. The websites were absolutely hopeless. I couldn't get any help anywhere. But the areas that I did find help were in some of the chronic fatigue ME association areas where they talked about being careful with your energy and spoon theory and not overdoing it and so on, and there were videos about videos were helpful because I couldn't read at the time. So I learned that I had to be careful with my energy, and I learned that physical activity, you have to add that together with emotional activity and also mental activity. So if you overdo all of these things, then you make yourself feel unwell. There seems to be a protective mechanism of doing something. And I learned about what is apparently called post-exertional malaise. And one day I felt much better, and I did 45 minutes of HIIT. And that made me feel very unwell the next day. So I started convincing myself I had a sort of chronic fatigue syndrome. And I started being careful about my heart rate, because there was something in the literature, in the ME literature, if your heart rate goes above 110, you will make yourself unwell again. So it was a really difficult time. So this went on to 6 months. I started writing to people because I was convinced that many people were overdoing it. I was a privileged individual at home, able to work from home. I was fearful of others who had to go out to work to earn their money, making themselves unwell because they were overdoing it. And I think actually there were a lot of people overdoing it in this kind of boom-bust cycle that goes on. And I suppose then what happened is I started saying, well, actually, you can't fight this. A friend in Australia said, you can't fight this, Paul. You have to do a deal with the virus. You have to accommodate rather than fight the whole time. And I have always been a fighter and a pusher. But the strategy with this illness is not around fighting it. You have to do a deal with whatever's going on inside your body in order to give you time to recover. I mean, since things started to settle down, the fatigue settled in fairly substantially, but the night sweat started disappearing around about 6 or 7 months. The brain fog was still there. The difficulty with speech was often still there. And I was lucky because I came across through my evidence-based networks a psychologist who really helped me basically understand my autonomic system had gone out of kilter and that I needed to use my conscious mind to kind of reset things. And, you know, I found it really helpful. I stopped monitoring my symptoms. I stepped away from the Facebook pages. I thought about many happy things in my life. I pushed out the illness with feeling better, not catastrophizing. And I'm now not 100%, but I have to be careful, but I am so much better. I would say I'm 96% of my 95% of what I was before. So just reflecting back on those on this more broadly in the last 5 minutes or so, this is happening everywhere. Globally, it's happening everywhere. I've had reports from India, from South Africa. I'm guess it's happening in the Philippines. People say that it happens in young people that appear to be, well, I'm hardly young, but people that appear to be fit. I think this may or may not be true. I think people that are fit will recognize impairment in their fitness and not being able to do what they could do before more readily and are more likely to talk about it. So I'm not sure about that. I think it's become clear that there are a whole series of different forms of long COVID. You know, I've got one that's particularly associated with fatigue. I think fatigue is common across all the syndromes, but other people have very disturbed heart rate issues, rapid heart rates that kick in spontaneously. Other people have continued crushing sensation and chest symptoms. So there are emerging symptom complexes, I think. There is also increasing research evidence on body scans and CTs and of myocarditis. People are particularly concerned about the myocardium. I am not up to date with all the literature on this, but I am an informed consumer. But my impression is that this is a virus and the post-viral syndrome that has never ever been so intensively investigated. I remain to be so many of the functional significance of what's being found needs to be observed to see whether people are permanently disabled or whether people recover. It's very common for people to think the virus is persisting. There's been some recent reports of the virus being found months later in the gut, and it's caused an existential crisis on the Facebook pages because the long haulers on the Facebook pages really were beginning to accept that this was a post-viral condition, and now they're sort of thinking the virus is still there. I really don't think it is for the bulk of us. It has long gone, and these are post-viral syndromes that we've seen, or infectious diseases doctors have seen over many years. And the catastrophic thinking that comes out of this between people that are unwell, I think, does them damage. It makes people obsess about their illness even more. So there you are. I mean, I said I'd talk for 20 minutes or so. Let's see over the coming years what happens. I honestly believe for many of this is a post-viral syndrome. I do agree that in the sort of 4 weeks to 12 weeks period, there may be persistent virus and funny things happening with other people. We've seen strokes and pulmonary emboli and so on in this age group. So it is a very odd virus that is causing a variety of symptoms. But you know, Epstein-Barr virus, people are ill for 2 years after it sometimes. Chikungunya, as I'm living in a house at the moment, doing convalescing with a young woman who had chikungunya, and 2 years after her chikungunya, she got quite severe arthritis in her joints. It was clearly related to the initial virus infection. So I think viruses do cause long-term conditions. It's just that we have never had this number of people and this number of sort of young mixed people of ages. So many people are disabled by this, needing support, needing psychological support, needing practical help. And as health professionals, we need to help them by reassuring them, giving them practical ways to improve, avoiding catastrophic thinking, and encouraging them to be kind to themselves, get their families to be kind to themselves, and that hopefully they will all recover. Thank you.
All right. Thank you very much, Dr. Garner. That was very, very, very interesting and very articulately shared with all of us. So, you know, in the Philippines, like every week, patients who finished already like 14 days of isolation are labeled as recovered. And actually, I don't know of any current post-COVID clinics that are open for patients who have had COVID. We sort of like discharge them. And like you said, we assume that they're going to be okay after the 14 days. Do you think this post-COVID clinics might be of help to patients who would be suffering or who suffered things like what you experienced?
I mean, I have a personal view on this, which isn't necessarily an informed policy view, because I have to be a little bit careful because I am a patient and I need to stay in my lines of areas that I can recommend. So I'm hesitant about making broad generalizations about policies around rehabilitation services in countries because I'm not an expert in that. But my own gut feeling is that by and large, many of these symptoms will be either dealt with within the home, within the individual, or will reach public or private doctors. So it'll be the first point of contact that people will go to. So I do think equipping in the UK, equipping general practitioners, and I think probably similar CADA in your country, with an understanding of what this long COVID can look like. So I think the first thing is not for specialist services, but to make sure that your general doctors and others are aware of the wide range of symptoms. And you know, because that in itself is quite important in their interaction with patients. So if you go in to a doctor and say, I'm getting twitching under my skin, you know, I'm getting fasciculation just here. I mean, you see on Facebook pages, people have actual locations where it keeps cropping up. And then the doctor doesn't realize that this is COVID. The doctor will say, oh, don't be so stupid, you're just neurotic, you know, you will be dismissed. I mean, Athena Akrami is a professor of neurosciences at University College London. She's top of the game, and she was a long COVID sufferer, went into work and said, I'm aching all over and I'm feeling really down. and her male colleagues, who were also doctors and professors, said, "Oh, you just pull yourself together. You're just depressed because of this illness, and because you're, you know, in brackets, because you're a woman." I mean, she was dismissed. It's reported in the Atlantic, and so this dismissal of symptoms does not help people. It does not help, and I think it has even more serious implications because it drives people out of the healthcare system and into the Facebook catastrophizing and into populations of people with chronic illness that then will unwittingly perpetuate this illness in others. So simply recognizing and listening to patients, and actually, you know, patients and the patient groups knew far more about COVID, and particularly long COVID, than the doctors did at one stage. So you were seeing the Facebook pages who had quite rightly interpreted that the antibody tests were often negative, and that was way before the doctors, many of the doctors had realized that the test was not terribly useful. So recognizing it is very, very important. Then there needs to be consideration, and I've lobbied for this hard in the UK, very clear indications of when to investigate and what to investigate. Now, if largely many of these things are going to get better, then doing whole body scans and MRIs and so on people will often not be very helpful. And of course, these tests are expensive, and if you've got a public health system, the people that will benefit are the shouty middle classes like me, and the poor people won't get access to them, so that it worsens inequity. But, you know, I mean, for example, when I had brain fog and difficulty speaking, there was a push to get an MRI. I kind of wanted one because I had a family history of cardiovascular, of brain hemorrhage. So I thought it'd be best to sort this out. But it showed nothing, and almost all the MRIs with people with brain fog and these symptoms show nothing at all, and is you know, generally would be regarded as a waste of money. So having very you know, people getting Marissa and yourself and the senior clinicians together in your country to consider what guidance should be given and when people should be referred and when people should be tested and have tests and what tests should be done would be a good thing, rather than it being a bit scattergun as it is at the moment.
Okay. Thank you, Dr. Garner. There are some questions in the Q&A.
Yeah.
So the first one is from Dr. John D. Buensalido. He says, "Thank you, sir, for sharing your experience. After your COVID-19 infection, do you still go out? And if you do, what is included in your must-wear PPE? Mask, eye protection, face shield? So, have you done some special protected socialization after your experience?"
Oh man, I was so even though I was really in a state of collapse for some of the time, I'd get out walking. So I think, you know, even at 4 weeks into the illness, I was walking, and I think it's very important to keep people out and about. I did consult quite widely about whether I was infectious or not, and I clearly wasn't. You know, I mean, the infectious bit is when you're much earlier on in the illness, so I am less cavalier about things. I don't catastrophize about getting another infection. Some people catastrophize about going out and they shut down. This fear you get is terrible. I am in an island of 110,000 people where they've had, I think, 30 cases of COVID in total in Grenada in the Caribbean, because they have such good public health control in the country. Everyone wears masks. I mean, everyone wears masks. You go into a supermarket, there's no argument. There's no argument. There's no debate. Everybody wears masks. Here they don't in the street. People respect social distancing, and this is in an island which doesn't have any disease, right? So people are very respectful of it. So, and I follow the good general principles as stated.
Okay, there's another question. Good evening, doctor. I contracted COVID-19 last July. I had controlled asthma before. However, at day 14 of illness, asthma became severe, needing GINA 5 treatment. However, all asthma workup seems normal. So can this be long COVID or COVID aggravating and asthma presenting as long COVID? He says, I feel you. It's really difficult. Thank you.
Okay, there's so many questions. I'm dependent on you for so thank you for doing that. I mean, I do, you know, as I say, I'm not clinically involved in treating people. We know that the COVID lung damage relates to probably things to do with blood clotting and the stickiness of the blood somehow combined with weird immunological things that are going on rather than a standard viral pneumonia. I can imagine someone with asthma then is going to be put at risk, and this is an additional, any kind of lung involvement is going to be an additional risk, but it may not be that COVID causes increased bronchoconstriction. But certainly the people I know with long COVID that have persisting symptoms seem to have this in the absence of clear pathological findings from CT, MRI, or chest X-rays of the chest. And each time I ask those people, do you think this has got an autonomic, a central control mechanism behind it? And I don't know. I mean, certainly the issue with SATs and people being severely unwell with really, but appearing to be okay with very low SATs is also a feature in actually in long COVID. There are a number of people that actually have are running quite low SATs, even though they're otherwise well, and I think that's an area for research. So I can't advise much further than saying I sympathize with you. If you've got asthma, I think I'd be very careful about it, but do consider long COVID can have long-term effects, both in damaging the lung and with potentially autonomic things that are interfering with your regulation of your breathing.
Okay, there's a friend here who asked a question from Mary Ann Langsang. Hello, Paul. Delighted to listen to you up close regarding your personal experience. Thanks much for your talk. Did crowdsourcing help in determining what coping mechanisms work best? Are you writing up, analyzing patterns of chronic fatigue from the millions of people you have written to?
So I thank you, Mary Ann, and I'm delighted to be here too. So in a sense, the Facebook pages sort of stepped in early on. You suddenly had a group of people that were, and in the UK, were all isolated at home. You know, nobody was going out. Everybody was scared to go out. Everybody was trying to make sense of this illness. The health professionals were concerned about the people dying, and all these people put on ventilators that they couldn't get off ventilators. So the communities were helpful in validating what was going on. I am unconvinced actually about how useful they are in identifying strategies, or if they are useful in identifying strategies, then you have to have a pretty strong filter on it. So let me give you an example. On the long COVID doctor site recently, so this is just doctors with long COVID. I mean, just doctors. I mean, it's people that have a GMC number in the UK. Somebody posted ivermectin and tetracycline, ! This is the cure for long COVID, right? And then there was loads of comments and yeah, I must try some ivermectin. I tried it and tetracycline looks a good drug. And I wrote, ivermectin in some laboratory studies has shown to have some in vitro studies, some activity against COVID, but you need to take it orally 1,000 times the dose that is normally recommended to have any potential for in vivo. And tetracycline is an antibiotic, and this is a virus, right? Please use the standards of critical appraisal that you would normally apply in your clinical practice to your patients, to yourselves. This doesn't make sense. God, I got such abuse. Doctors are useless at treating themselves, you know, and that's why I've been very circumspect about being careful about my own diagnoses. So, and what tends to happen with this illness, and this is another observation that I've had, is because of its random nature, you don't know when it's going to come back, right? You can be fine one day. I'm fine almost all the days now, but you could, you know, when I was ill, I'd be absolutely on top of the world, almost over on top of the world. I was always high on top of the world. And then the next day I'd be bang, rotten, ill and sweaty and terrible. And you couldn't predict where it was coming from sometimes. But what the Facebook group and other groups do is try and suggest there might be something that's precipitating it. So you get into this mindset, oh my God, what's precipitating it? And I was at home and a friend had come round and I was feeling quite good on a Sunday, and they brought cake and then they left, and most of the cake was left. And I hadn't had cake for ages, and I ate 3/4 of a Marks and Spencer's Victoria sponge. Bang, I ate it down all at once. And then I felt terrible the next day. I thought, oh my God, maybe it's sugar intolerance. You know, I put it on the Facebook page. They said, oh yes, it's very well recognized. People, your sugar load will precipitate your illness. And so I became convinced I was allergic to sugar. This is such nonsense. This is such nonsense, Regina. And you know, and then the next time, it was my birthday and I had some little cookies. Somebody made me some little cookies and I thought, oh, I better not eat those because I'm allergic to sugar. And then I nibbled one, and then the next day I thought, well, maybe I feel a bit unwell. Maybe I am allergic to sugar. So they can set up these cycles that I think can be quite, and that's why some of the identity of these long COVID patient groups is very important for validation. But I think on the treatment side, they can result in anecdotal acceptance of things that really don't make any sense. And then these things through Pavlov conditioning can become self-fulfilling. So yeah, so that I think it's got pluses and minuses. And if anybody was ill, I would say get into the Facebook things early on. Later on, drop out and try and look after yourself or work with medical practitioners and get yourself better.
Okay, so I have time for 2 more questions. There's a question from Dr. Janice Kowili. Thank you, Dr. Garner, for sharing your experience. I was struck by your statement that we should be patient with our COVID-19 patients. I agree. I have a question regarding long-haul COVID-19. Do you think it's consistent with the post-COVID multisystem inflammatory syndrome? Is MIS a spectrum of illness?
Yeah, I think it is a multi-system disorder. It's a post-viral syndrome. There's sometimes advantages with labeling things. I think if I posted on Facebook to people that I was encouraging, or the people that I talk to quite a lot of people that are unwell with COVID, just to share experiences, because often that can help. And if I said to them, you have, long COVID, you realize this is a multi-system inflammatory syndrome, then bang, it kind of it helps perpetuate this catastrophic thinking. So I don't agree or disagree with that really, but I did talk to someone recently who said that they were doing really, really well. They'd had 1 or 2 setbacks, which happens as you with this illness as you're resolving it, and then they read that COVID remained in the gut, that the virus had been found in the gut, and they went into a downward spiral. So I do think this avoiding, and I think the doctors make the errors with, I've still got a fever, therefore it must be a virus in my body. No, post-viral syndromes, immunology often give you this. Autonomic disturbances will mess around with your temperature. So try and avoid getting into lots of biological explanations. Is it interleukin? Is it this? I don't know. And nobody knows. But the prospect of it sometimes doesn't help people use their mind to overcome what their misfiring autonomic system.
Okay, this question requires just a short answer. Do you have data on how many % of these COVID positive patients who recovered from the illness developed the long haul COVID syndrome? Is it more common in older age groups?
So there is a group that we're working with, as a call later today, trying to summarize and systematically review that. The problem has been really having prospective cohorts, because people like me I'm not even on the statistics of the UK of being a COVID case. I'm nowhere. I'm not a case. So no, and Phil Spector's app in the UK has been useful, but is subject to many people dropping off, particularly when their symptoms persisted. They just got fed up with filling it in. So I think it's a few percent. But if you look at it in terms of Facebook and in terms of talking to neighbors, I've got, in my street, I've got 3 people at home, all with long-term symptoms of COVID, and that's what I know about. So there are friends of children that are taking time off school with long COVID. So it is extraordinarily common, but I would be hesitant of giving an accurate statistic, because I think we need to review the definitions and now it's been looked at. But it's common.
Okay, so we have no more time. We ran out of time. Certainly, this is an eye-opener. The most important thing that I think Dr. Paul Garner taught us today is that we need to recognize that this is fairly common enough and we need to be aware of it and be patient to our patients. Okay, so I'd like to present the certificate of appreciation to Dr. Professor and Dr. Paul Garner for taking time to be with us today and for your excellent lecture. Thank you very much, Dr. Garner.
Thank you. I'm very honored to be back with you and amongst my friends there in your country. Thank you for asking me.
Thank you. Thank you very much. And we'd like to move on to our next very important topic on vaccines for COVID-19. To introduce the next session would be Dr. Jodor Lim and Dr. Brian Lim.
Okay, good evening, everyone. Good evening and good morning to some of our friends. Thank you very much, Nina and Professor Garner, for a very interesting talk. Well, today, the buzzword is vaccine, vaccine, vaccine. When will we get our vaccine shots? Which vaccine is the government procuring? When do we have this? So all of these are actually words that we ask ourselves now. We're almost near the anniversary of the COVID discovery, and the vaccines already being tested. Some have had the preliminary results already. So tonight we'll have a very special talk on vaccines, and we have invited a person who I heard lived in the Philippines before, and probably Soumya can share with us some of her experience when she was here in the Philippines. So we have tonight talk on vaccines, and with me is Dr. Brian Lim as our co-chair. So our speaker is the chief scientist at the World Health Organization. She leads the science division, which ensures that WHO harnesses the power of science and innovation to improve global health and deliver impact at country level. She was formerly the Deputy Director General for Programs at the WHO from 2017 to 2019 to share with us vaccines for COVID. Let's welcome Soumya Swaminathan. Soumya?
Thank you, Dr. Jodor Lim and Dr. Brian Lim, and thanks also to Marissa for having invited me to join your conference today. Dr. Marissa is a member of our executive group of the steering committee for the Solidarity Therapeutics trials and has helped us over the last several months to run these multi-country, multi-arm adaptive trials that we can talk about later. But right now, I think the topic that we're discussing is vaccines, and I'm going to try and share my screen with you all. How do I share my screen? Okay, let me try. Slideshow. Okay. I hope it's going to work. Are you seeing anything? Are you seeing my slides?
No, it's blank, Soumya.
Yeah, I don't know why it went blank when I said slideshow. Oh dear.
Naka-Teams siya.
Okay, that's not good. Let's try again. Okay, even if it doesn't go on slideshow, I think we'll just take it. It's maybe the network. What I'll try to do today is to maybe just have an overview of where we are with vaccine development for COVID-19. And as you know, this has been a very, very exciting year in terms of the science. And that's why it's been so interesting for me, because as the chief scientist at the World Health Organization, I have obviously been working very, very closely with groups around the world, with scientists, with the vaccine developers, with therapeutics developers, with member states as well, trying to advance the development of vaccines as well as diagnostics and therapeutics. And I'll, during my presentation, share with you a little bit about what the role of WHO has been. I see that somebody has projected my slides. I think that wasn't me, it was the organizer. So could we go to the next slide, please? Okay, and the next one. The next. Just starting with a snapshot of the epidemiological situation so that we remind ourselves where we are today. And on this graph, you can see from the beginning of the year till now, how the epidemiology has progressed. And the different colors on the graph actually are the different WHO regions. So if you see the yellow, this is the region of the Americas, North and South America. You can see that they really, over the summer, really had the maximum burden of disease. And then there was a slight slowing down, but again, they are the ones, the North and South America region is contributing to the largest number of cases. You can see that in the last week, we had 4 million new confirmed cases. You know, the first month when it started, we had a few cases a day, a week. And then in the month of May and June, it used to take 1 month for us to reach 1 million cases. And now we're at 4 million new cases a week. So you can see this, the exponential growth of this pandemic. Similarly, if you look at the black line here, these are the deaths. And there was a peak in March and April with a spike in deaths. This came down and more or less stayed at a plateau for many months. And then you see again in October, the deaths climbing. So from 5,000 deaths a day over the summer months, we today are at 10 to 11,000 deaths a day. So you can see 70,000 new deaths confirmed over the last week. Cumulatively, we are approaching 65 million cases, and we've crossed 1.5 million deaths. Now again, these are confirmed cases, confirmed deaths. We know that in most countries, the statistics take a long time to get updated. It's not real time, and therefore we will not really know the true mortality due to COVID for some time. And also we will not know the true mortality, excess deaths due to other diseases which were impacted by all the actions that countries had to take this year. On the right-hand side, you see listed the top 10 countries with the highest number of new cases in the past week. The graph also shows you whether it's still on the ascent, whether it's declining or stable. Next slide, please. So really the point is that we are not nearing the end at all. We are at a very critical stage, especially with the winter in the Northern Hemisphere, when people are likely to be more indoors, and we know this virus likes to spread in closed, unventilated environments. Now this graph is a pictorial to see by the shade of the color where you have the maximum number of new cases being reported in the last 7 days. This is per million population. Again, the darkest shades of red are in Europe and in the Americas. Africa, you can see, has been in a much lighter shade, though in some countries in Africa, there has been an increase in the number of cases over the last couple of weeks. But if you look at it per million, it's much less. You can see also Philippines there, which is sort of like the other Asian countries in the medium range. Next. The next graph basically shows us the deaths. Again, it's per million population, so it's corrected for population in the last 7 days. Again, highest burden in Europe and Americas, but also in parts of the Middle East, and Russia has been seeing increasing cases and deaths. Now, what happens is cases increase and then deaths follow a couple of weeks later. There's always a lag period. Next. The next slide, now we move on to the science behind the COVID vaccines. The next slide. First, we need to understand the immune response, because obviously without understanding the immune response, we do not go about developing a vaccine. Now we know fairly well. Of course, there have been 7 coronaviruses in the past that we know of. 4 of them really cause upper respiratory infections. They account for 15 to 30% of upper respiratory infections. And then there are 2 coronaviruses which have been causing severe disease in the past, the SARS-CoV-1 and the MERS-CoV, which still comes up from time to time, mainly in the Middle East. So we knew a little bit about the immunology of those diseases, but in this particular virus, it's taking time to understand because obviously it's a new infection. Now, if you see here, what happens is that there is an incubation period. We know 0 to 14 days. Most people develop symptoms around day 5 to 7. The virus peaks just before that. So the virus starts peaking just before the onset of symptoms. So that is why it's so important for everyone to take precautions, because people are infectious in the pre-symptomatic period, and then it very rapidly declines. And by about 10 days, you do not get viable virus being excreted, even though the RNA may be detectable for longer. We know that IgM antibodies start getting produced towards the end of the first week, and they peak around week 2 to 3 and then decline. And then you have the IgG antibodies, which appear only after 10 to 15 days, and they achieve a steady state, say around day 21 to 28, and then they are detectable longer. How long? This we still are finding out. Some studies the cohorts are being followed. Many studies have reported a waning of antibodies. So if you test people beyond 4 months, there is a waning. However, studies which have looked very carefully and which use sensitive assays are able to detect antibodies even up to 6 months. And those studies are continuing. Now, we know that there are memory B cells as well. So we shouldn't be too worried if antibodies are not detectable, because the memory B cells can come into action as soon as it's stimulated when it sees the antigen again. Now, apart from all of that, of course, we have the innate immune response. Now, the innate immunity is very important for the initial response. You can see that's the one that kicks in. And that is the one that secretes interferons and is effective against most viruses. And innate immunity correlates with a number of factors, the most important of which is aging. But immune aging is not necessarily the same as biological aging. So what we call senescence of the immune system can occur earlier in some people with comorbidities. It can occur in others as well, maybe we don't understand it fully. So while biological age is a good marker of immunological age, it's not always that the two go together. We need to remember that. People who have good innate immune responses, children, means that they have a lot of naive T cells in their system, which are the ones which come into play immediately. That's why children seem to suffer less from symptomatic disease. This declines as we age. Cellular immune responses, again, something we're beginning to understand. We know that it's been important in SARS and MERS infections. So T cell responses are probably playing an important role, particularly in the longer term, the memory, and when there's a reinfection, it's the T cell immunity that might be playing a bigger role. So these are all important, because when you develop a vaccine, you need to understand what kind of immune response are you trying to elicit and what would be the correlates of protection. Now, this we don't know yet. We will know soon, because with all the vaccine trials that are being done, measuring different immune responses, we will be able to get an idea of what are the markers that correlate with protection. And that's important because future vaccine trials may be able to use correlates of protection using immune markers rather than following people up for development of disease. In many diseases, we still don't understand the correlates of protection, and TB is a good example. And that is why it's so difficult to develop and conduct vaccine trials for TB, because you need to follow people for the development of TB disease as an endpoint. There is no marker that we can use. If we can go to the next slide. And the next one, please. Now, in an immunized, immunity obviously can be acquired naturally after you get the viral infection or after vaccination. And what happens is that you get initially a cellular response, and at the cellular response would continue. So if you stimulate T cells with these antigens, you should be able to elicit an interferon gamma response. Either can be measured through flow cytometry or through ELISpot assays. The antibody response obviously then would last. Next slide. And that's what we think would actually help protect people from the next bout of infection. Now, again, we've gone through this humoral immunity, cellular immunity. So I think we can move on to the next slide. Now, vaccines, how do they work? They deliver an immunogen to the immune system so that immune system gets trained to recognize it when it is presented with the natural pathogen. And again, it could be through neutralizing antibodies, which are produced by B cells, and CD8 T cells and CD4 T cells, which also recognize viral proteins. Now, the way that the immunogen is presented to the body, the route of administration, would be important, both for the kind of immunity it elicits, because some types of vaccines elicit better T cell responses than other types of vaccines. It's important also for age-related responses, the duration of protection, and the side effects. Side effects could also vary by the type of vaccine that we use. And we know that in the COVID vaccine development, people are using practically every platform that is available, starting from attenuated pathogens. So we use a live replicating. You can use live non-replicating, like the Chadox vaccine and like some of the other adenovirus-based platforms. A very common way is, of course, to kill the pathogen and use killed. And this is an old model of developing vaccines. Many of the Chinese vaccines under development are using this killed approach. Subunits, you take just a part of the protein, which either derive from the pathogen or you can create it. Recombinant, you generate it in a host cell and purify it, like the Novavax vaccine. You can use peptides, synthetic fragments. You can use vectors, the adenoviruses we mentioned. And most novel are the two DNA and the RNA platforms, which have been tested for development of vaccines in humans. So there are a number of other mRNA vaccines which are in clinical trials for diseases like chikungunya and Lassa fever, but none have actually been gone through a phase 3 trial or deployed. And today you would have heard the news that the Pfizer vaccine has been given approval by the MHRA and will be rolled out very soon, say within the next few days in the UK for the high-risk groups over there. So first time that we'll actually be using an mRNA vaccine on a larger population. It has been used in phase 1 trials, so there is some experience of its use in humans. Now, the DNA and the RNA vaccines obviously carry the message into the cells to create the spike protein within the human body. All the others, we inject the spike protein in one way or the other, or you're injecting the whole inactivated virus. But here, you inject only the instructions to create the spike protein. It's produced by human cells, and then the antibodies and immune responses are also happening within the body. Next slide, please. It's also important about systemic immunity and mucosal immunity. Respiratory virus, you would like to have also mucosal immunity, and that there are under development now vaccines which are trying to do it through the nasal route, through spray. There's one that's on a measles platform, and there's another one which is combined with an influenza vaccine. That would be perfect if you could get a vaccine that combines flu and COVID-19. I mentioned how these nucleic acid vaccines work, either DNA or RNA. The issue with the DNA is that you need a special device to administer it. It's called electroporation device. It's electric pulses to enhance entry into the cells, and then it has to be converted into mRNA. But for mRNA vaccines, you can inject it directly. But the issue with the mRNA is it is not stable. That is why it has to be encapsulated with a lipid nanoparticle layer. And that is why also it requires storage at -70. At least the Pfizer one does. The Moderna one can be transported at -20 and stored in the refrigerator for up to a month. Advantages for these vaccines: strong cellular immunity and rapid production. Obviously, you know, these vaccines were produced within a couple of days of the genetic sequence of the virus being made available by the Chinese scientists on January 11th. By 13th of January, Moderna had actually developed a candidate vaccine, and third week of March, it went into human beings. So overall, it's taken us 300 days approximately to go from a genetic sequence of a virus to a vaccine being approved in the UK today. 300 days. It's a record. That's advantages. You can also produce it very rapidly, as I said. Disadvantages: antibody responses may be lower than other technologies. That's theoretical. Now we need to see in real life, and we've seen the results from both Pfizer and Moderna showing above 90% efficacy. Of course, very short term. The follow-up at this point is hardly 2 to 3 months, and that's not enough for us to really make a judgment call. We will need longer follow-up, and that is why we are very keen that even as these vaccines get emergency use authorization, that trials should continue, that blinded follow-up should continue as long as it's ethically feasible, and that these cohorts need to be kept under follow-up, both for efficacy and longer-term safety. Next. Till date, as I said, no nucleic acid vaccine has been approved for use. Now, we know that most people who recover from SARS-CoV-2 infection have antibodies, and these levels of neutralizing antibodies are higher in patients who had severe disease and much lower. And in about 10 to 15% of cases, mostly those who have asymptomatic infection, we are not able to detect these antibodies. That doesn't mean they don't have the memory cells. Now, when you do an ELISA, you don't really know what type of functional antibodies you're measuring, whether they are neutralizing or not. So ELISA tells us about the quantity, not necessarily the qualitative aspects. Next slide. With MERS, also the same thing was noticed, that the patients who had more severe disease had higher levels of neutralizing antibodies, and those are detectable for several years. In fact, when they looked at SARS-CoV-1 patients, it's now 17 years, it was 2003, there were still T-cell responses to the SARS-CoV-1 antigen among people who had recovered. So that means that memory cells have lasted for many, many years. How long does it take to develop a vaccine? Well, if you had asked the same question 6 months ago, the answer would have been normally 10 to 15 years. And the shortest time it's taken for a vaccine has been the mumps vaccine was developed in 4 years. And I think Zika also took around that time. But before the Zika vaccine could be widely tested and used, the problem was over. Now, how has it been possible for COVID-19? Because the steps have been done in parallel, not in sequence. So why phase 1 already rolling into phase 2? Regulators have been looking at the data continuously, providing approval to go from phase 1 to 2 to 3 very rapidly. There's also been investments in manufacturing made right at the beginning, not waiting for the results, and a minimum amount of safety data. So FDA and other regulators said we will be satisfied with 2 months safety data for at least 50% of the patients who have been enrolled in the phase 3 trial, along with efficacy data, and then we will need the post-EUA studies to continue. Next. So there is a need for a large number of post-introduction studies, especially around safety. So pharmacovigilance is really important to prepare for. Next. Now, we don't need to go into why we use vaccines. I think this audience knows that. Let's move on. The only point here really is about the herd immunity, and a lot of discussion and debate in the people about herd immunity and how it can be acquired. There are people who are pushing for naturally acquired herd immunity. That's not possible. As you all know, there's no infectious disease for which you get natural herd immunity. It cannot happen unless you allow the infection to keep on spreading and making people ill and killing people. That's not a good way. So when we talk about herd or population immunity, we should only talk about it in the context of vaccines. But we need to explain this to the public. So I think key here also is our communication, and you're all experts. You will be called upon to communicate, talk to the media, talk to people about, especially the new platforms. People are worried that these mRNA and DNA vaccines can do damage, that they can go in and change your, genetic constitution of our cells and so on. Obviously, that's not the case, but we need to reassure people about these platforms as well as about the process of testing and how data is going to be collected and used. Now, I want to say a word about the R&D blueprint, because this is something that's really played a role, I think, in this outbreak. Many of you would be familiar that after the Ebola outbreak of 2014-15, WHO was asked to create a blueprint to prepare for R&D during emergencies and outbreaks. So a lot of experts met at that time. They not only defined this global strategy for what needs to be done, but they also created a list of priority pathogens. And on that list of priority pathogens was a disease called Disease X. Next slide. And Pathogen X. And today, that's what we are dealing with. We are dealing with Pathogen X. And R&D blueprint framework actually laid out what you need to do. So right from creating a roadmap to developing target product profiles for vaccines, diagnostics, and therapeutics, to defining regulatory standards, to designing, putting up core protocols for vaccine or therapeutic. Because when you are in the middle of an outbreak or an emergency, you cannot start to do all of these things. That's why it needs to be done in advance, and the world needs to be prepared, and every country needs to be prepared so that you can actually start the research when the first case is identified of whatever the disease is. And we've done this for Lassa, we've done this for Nipah, we've done this obviously for Ebola as well as for other hemorrhagic fevers, and we're also working on chikungunya. And now this year we've been focusing on COVID-19. Next slide. And for outbreaks like Nipah, when you have small outbreaks which are contained quickly, it's important to have an accumulation of data over many outbreaks. So this is where you also need collaboration between countries, either within a region or across the different regions, so that today the outbreak is in Bangladesh, tomorrow it's in Malaysia, the after it's in India. If you have a common protocol, if you have an agreement, then even if there are only 20 cases in a country, you can quickly do the testing of therapeutics or diagnostics or whatever it is and accumulate data over a period of time. For rare diseases, this is important. So the R&D blueprint, we held an innovation and research forum in February. We came out with a research roadmap that identifies, the next slide, the knowledge gaps and activities that are needed, priorities around 9 thematic areas. This included vaccines and therapeutics, but also included things like clinical management, transmission, infection prevention and control, social and behavioral sciences, and then looking at the human-animal interface. Next. For vaccines, what did we do? We have been maintaining a landscape of COVID vaccine candidates, which is constantly updated. We get information from all the developers, and we provide this on our website. We've put up criteria for candidate vaccine prioritization. We develop target product profiles so that developers know what is the minimum requirements, what's the benchmark for an ideal vaccine, and what is an acceptable vaccine. So we said for an ideal vaccine, we'd like to see over 70% efficacy. We'd like it to be a single dose. We'd like it to be very safe. We'd like it to be stored at room temperature or in a refrigerator. We'd like it to give lifelong immunity and safe in pregnancy and so on. But then you can't always have the ideal. So we also have what are the minimum or critical ones that we would need to look at. So we said minimum of 50% efficacy, could be 1 or 2 doses, immunity for at least 12 to 24 months, storage maybe -20 or refrigerator, so on. So we have that. That's been very useful for regulators also to say, okay, these are the benchmarks that a vaccine needs to be assessed against. Then we have the operational and design considerations for human challenge studies, and we also have a document on what are the ethical issues and an ethical framework if someone wants to get into human challenge studies. We had expert groups working on animal models, on the assays, because you need to standardize assays. Every vaccine trial needs to use a standardized assay, otherwise you cannot compare apples and oranges. And then, of course, we had the protocol. We developed what's called the Solidarity Vaccine Protocol. It's a phase 3, multi-candidate, head-to-head, adaptive, multi-country type of design, which we are now working towards rolling out. And the Philippines has been very, very actively involved right from day one in not only Solidarity Therapeutics, but also in planning for the Solidarity vaccine trial. It's taken us a little bit of time because most of the early vaccines, the companies have launched their own programs. What WHO wants to do is to support all or as many vaccine candidates as possible to undergo good phase 3 trials, because we need a number of vaccines. We need vaccines for the developing world. We need affordable vaccines. We need vaccines that can be stored at room temperature or in the refrigerator. And therefore, we need to encourage the development of more candidates. And that's where we're looking at Solidarity, helping those candidates to go forward. Next slide, please. And again, the advantages of doing this type of big global study is you can evaluate several different candidate vaccines, against a common placebo arm. So you're saving time and it's efficient. You can enroll patients across the world in different countries. Some countries would have a big outbreak, some countries would have slowed down. If you have spread sites across the world, you can continue to enroll people even where the epidemiology is very high. Even if other parts of the world, it has gone down. Like look at China, for example, they controlled it. So the Chinese candidate vaccines need to go out of their country. They cannot do the trials within their own country. Again, it's efficient. You get results fairly rapidly because you're enrolling in high incidence sites. And of course, it fosters international collaboration and hopefully equity of access. Next. I'll try to go fast. We need many vaccines. I've already explained why. First of all, not all vaccines in early development will be successful. Generally, only 10 to 20% are successful. But let's see, the first few ones have been giving us very positive results. Let's hope that continues. Next. Okay, a couple of words on emergency use listing. I mentioned that this could happen. Every national regulatory authority has the right to give an EUA for a particular product, okay, based on their own rules and regulations. The WHO EUL, we put out the criteria on our website. It's basically a risk-based procedure for assessing and listing unlicensed products in order to expedite the availability to people affected by a public health emergency, because this is many global agencies like Gavi and UNICEF will only procure those products which have either WHO pre-qualification or emergency use listing. And it also provides member states with data on the safety, on the efficacy, as well as on the quality of manufacturing of a product. Next. So many companies are now talking to the WHO about submitting their vaccine dossiers, either for an EUL and then later on for pre-qualification. Now, the criteria are, it has to be an emergency. It has to be a disease, obviously, which is causing a lot of illness. The vaccine candidate must have undergone phase 2 or phase 3 studies and have received an authorization from a reference NRA. There needs to be a data submission which follows all of the requirements.
Hello, everyone. We seem to have we seem to be experiencing technical problems. So
let's use this time for you to type your questions under the Q&A portion of the Zoom. So kindly type your questions while waiting for our speaker to come back.
Sorry, I got disconnected. Are you able to hear me?
Yeah, we can hear you, Dr. Swaminathan.
I will stop the I wanted to. So it may be more interesting to now have question and answer. Thank you.
Okay. So a very good evening, Dr. Swaminathan. Thank you for that very comprehensive lecture. So we have a couple of questions here. So I will lump similar questions together. No? So a lot of the questions here are on safety. No? One is, it normally takes 10 to 15 years to develop a vaccine. What are the measures to ensure that these vaccines are safe, considering the rush processing, so to speak? And the next question is it safe to give these vaccines to immunocompromised patients? Are there any studies to support this?
Yeah, these are very good questions. And on immunocompromised patients, obviously, these are the people who are at risk for severe COVID-19 disease. So they would be a priority group. The issue will be how good an immune response do they get. Now, as we get more data from the clinical trials, we will be able to understand this much better. Right now, we may not have an answer, though all of these vaccine trials are trying to enroll patients of different ages with different comorbidities, etc. On safety, as I mentioned, the reason for this rapid emergency approval is based on the urgent public health need for a vaccine. And that is why the regulator agreed to do with a basic safety data. So we need 2 months of safety follow-up data for people in the trial. And with that, they can apply to the regulators, and they can also apply for WHO pre-qualification. Now, this is only an emergency use authorization. It is a conditional approval. And the condition is that the manufacturer must continue the trial, must continue the follow-up, and must submit the data before a full license can be issued. And that usually will happen over the period of 12 months. At any time, if there are signals from the ongoing studies that are reported to regulators, the emergency authorization can be withdrawn. So I think this whole issue is really weighing the benefits and the risks. And this is the job of the regulators, to weigh the benefits and the risks. And if a vaccine is going to be given emergency use authorization, it means that the benefits probably outweigh the risks. But we know that many of these vaccines are going to need a longer follow-up in larger populations before we start seeing, signals of rare adverse events, which you may not see in the first 20, 30,000 people, but you start seeing it only when hundreds of thousands and millions of people get vaccinated. So the next year or two, there will be strong pharmacovigilance programs in all countries, and WHO will be keeping a close eye on this.
So thank you. You mentioned about weighing the risk and the benefits. There's a question here regarding the approval of the UK government to purchase and roll out a vaccination program next week. What are your thoughts on this, Dr. Soumya?
I think it's the same thing. The UK government is struggling with an outbreak which is really quite bad and quite severe. They're seeing rising deaths. Their NHS, the hospitals are overloaded. The health system is under strain. They want to do everything possible to prevent deaths, particularly among the elderly, among the nursing care residents. And so for them, that's again the benefit risk equation that they've looked at. And having looked at the data from the Pfizer trial, of course, it's limited in time, but the efficacy, again, the efficacy 94% is over a limited period of time.
Yeah.
And we don't know that efficacy will continue, but we're at a stage where countries and people are willing to take that risk because of the situation they find themselves in.
Yes.
It's an unprecedented situation, and therefore people are making decisions which would not normally happen for any other disease.
Okay. Dr. Soumya, I have 3 questions for WHO. Are there vaccine candidates already for the Solidarity vaccine trial? And how will WHO ensure equity in vaccine distribution?
So again, 2 very good questions. For the vaccine trial, we have a committee, a scientific independent expert committee that looks at the dossiers that are submitted to us from the different developers who are interested to be part of Solidarity. And they go back and forth with the manufacturers asking for more data and try to prioritize which ones are the most likely on how to proceed. So we expect very soon that those decisions will be made, and so that we can start, even if it's 1 or 2 candidates to begin with, and we can add more later. We do, as I mentioned, want to look at the 2nd generation of candidates that are now coming out of phase 2 trials and trying to get into phase 3. On the equity, this is the reason we launched the ACT Accelerator. The DG, Dr. Tedros, and many heads of state, as well as the heads of other global agencies, got together at the end of April and launched what we call the Access to COVID Tools Accelerator. And one arm of that is focused on vaccines. The others are focused on diagnostics and therapeutics. The vaccine arm we call COVAX. And you might know that we have now something called the COVAX facility that's hosted by Gavi, the Vaccine Alliance. WHO, CEPI, and Gavi jointly created COVAX. That's the facility that's actually now got 187 countries have joined. So it represents over 90% of the world's population. 92 of those countries are what we call the AMC countries, which will be provided vaccines at a very low cost or free. And the other 94 are countries and economies which are self-financing. They will buy from the facility, but they're going to pay for vaccines. So the goal is we have enough vaccine laid, 2 billion doses of the vaccine by the end of 2021. That's enough to cover 20% of the world's population. And 20% will take care of the most vulnerable and high-risk groups. So that will start blunting the impact of the pandemic. Then in 2022, countries will scale up their vaccination programs to cover much larger segments of the population. But we feel even if you cover 20% initially, the elderly, the high risk, the frontline workers, the healthcare workers, you will start seeing a good reduction in mortality as well as a protection of the health system. Now, it all depends on whether we can raise the funds. We've raised $2 billion. We need to raise another $5 billion to be able to meet our goals of 2 billion doses by the end of 2021. On timelines, we think we will have 500 million doses in the first half of 2021. So countries will start getting shipments of vaccine beginning in the end of the first quarter, maybe end of February, March, April onwards, but limited quantities. In the second half of 2021, hopefully there'll be larger quantities of the vaccine coming to countries to start vaccinating their priority groups. Right now, we're in the process of making deals with companies. We've already got deals with AstraZeneca, with the Serum Institute of India, which is also manufacturing the same vaccine, with GSK, Sanofi, and with Janssen. And we are currently in discussion with Pfizer and Moderna.
Thank you so much. And indeed, it's quite a challenge to roll this out. And it's quite comforting to have all of these equity measures that are being considered. So for one last question in relation to that, have you heard of any significant activity from the anti-vaxxers at this point in the pandemic? So, because here in the Philippines, we have quite a growing, considerable force of anti-vaxxers, also especially in the provinces.
That's an excellent point. Yes, indeed, we are very concerned. In many countries, we have seen that the anti-vax sentiment has been quite high. And I think partly it's people, not necessarily anti-vax, but people who are genuinely uncertain, like the question about the safety, the question about how did you develop these so rapidly when normally it takes 5 to 10 years? Is it safe to take? I think people have genuine questions, and we need to answer those questions and reassure them. The first set of people are going to be healthcare workers, frontline workers, of course, the elderly. Now, these are people who generally understand a little bit more, would have better health literacy. So maybe people will get confidence when they see doctors and nurses and pharmacists and lab technicians taking these shots. But I think the important issue that you raised is an issue of communication, is an issue of management of people's perceptions, and a small, I would say it's a minority who are really anti-vax, who are going out and really creating a lot of misinformation and spreading all sorts of rumors. So every government, I think, needs to assess the situation in their own countries and needs to start communicating with the public in an open and transparent way. I think that's the only way in which we achieve these high rates of vaccine coverage that we want to.
Thank you so much, Dr. Swaminathan, for enlightening us. At this juncture, may I request our chair, Dr. Jodher Lim, to present the certificate of appreciation.
So Dr. Soumya, thank you very much. So in behalf of the organizing of the Philippine Society of Microbiology, we'd like to present the certificate and developments of vaccines in the future. Thank you very much.
Thank you. Thank you so much. Appreciate it.
Okay. So at this juncture, we're going to our last plenary for today, entitled Diagnostic Utility of Saliva Specimens for COVID-19, to be chaired by none other than our past president of the PSMID, Dr. Julius Laciones, as well co-chaired by Dr. Gemelin Garcia. Good evening, doctors.
Thank you so much, Brian. I hope you can hear me loud and clear.
Yes, we can.
Good evening to all PSMID members, colleagues, and participants to this last session of the day. I welcome you all to plenary session number 9, which is about the diagnostic utility of saliva specimens for COVID-19. I will be chairing this session, and my co-chair, as mentioned previously by Brian, is Dr. Jem Garcia, a colleague from RM and Asian Hospital and Medical Center. Please note that our more than 700 participants has not really left us. They're still here, and I can understand because this next topic actually is very interesting. We know that the nasopharyngeal swab is the standard diagnostic tool, as it has been reported to be accurate and sensitive towards detection of the coronavirus. We know that it is inconvenient to many patients, and that is one of the fear why they don't want to be swabbed. Testing of saliva specimens is now being considered to aid rapid detection because the collection and its testing are relatively simple cheap and safe for both patients as well as professionals. By the way, Dr. Jem Garcia will be presiding over the Q&A portion later on and will officially close this session. Our speaker is a postdoctoral research associate at the University of Illinois at Urbana-Champaign up to the present time, where she also led the research and supervises the research for COVID-19 diagnostic testing. She was a postdoctoral scholar at the University of Chicago from 2014 to 2019. She completed her Doctor of Philosophy in Microbiology and Immunology at the University of Illinois at Urbana-Champaign. And most recently, she led a team of scientists to develop a saliva-based molecular testing of SARS-CoV-2. So please join me in welcoming our speaker, Dr. Diana Ross Ranoa. Yes, Dr. Ranoa, you can proceed and give the lecture, please.
Good evening. Thank you for having me as one of the plenary speakers for the 42nd Annual Convention of the Philippine Society for Microbiology and Infectious Diseases. So tonight, I will be telling you about one of the largest mandatory COVID-19 screening efforts performed at any university using a homegrown test that's developed here at the University of Illinois at Urbana-Champaign. So the title of my talk is Saliva-Based Molecular Testing for SARS-CoV-2 that bypasses RNA extraction.
I think we cannot
hear.
We
all have heard in the news
Can
about
somebody fix the...
one of the largest mandatory COVID-19 screening efforts performed at any university using a homegrown test that's developed here at the University of Illinois at Urbana-Champaign. So the title of my talk is Saliva-Based Molecular Testing for SARS-CoV-2 that bypasses RNA extraction. So we all have heard in the news about skyrocketing cases of COVID-19 in the USA this winter season. So looking at this graph from the Johns Hopkins University website, the green line corresponds to confirmed new cases here in the United States. And in this month of November, we already are averaging greater than 150,000 confirmed cases per day, way higher than any other countries in the world. In the state of Illinois, our 7-day moving average is right at the range of about 10,000 to 12,000 confirmed new cases per day. And our positivity rate is right at 12.2%, which is very high. So why is SARS-CoV-2 difficult to control? So currently, the Center for Disease Control, or CDC, recommends that only those individuals who are showing symptoms or those who have been exposed to individuals who tested positive for the virus should go through COVID-19 screening or testing. However, as shown here in this figure, about 50% of the spread, if not more, is from asymptomatic or pre-symptomatic cases. So upon infection, the virus incubates at an average of 4 to 5 days before symptoms onset. And shown here in red curve, which corresponds to virus shedding, an infected individual can start shedding the virus and infecting other people at about 2 or 3 days before symptoms onset. So obviously, testing and isolation of symptomatic individuals is not enough to control the spread. In addition to that, we also need rapid return of results. We want to know the results within hours and not days. And it's also critical to utilize tests that can be repeated multiple times per week. The second reason is that virus spread through salivary or respiratory droplets and aerosols. So if you have been in close contact with an individual that is infected with SARS-CoV-2, you can be infected with the virus through droplets in saliva and also in aerosols that contain the virus and can remain floating in the air in indoor spaces. And the other route of infection is through fomites, where the virus stays in several different types of environmental surfaces, and if you happen to touch those surfaces and put your hands in your eyes, nose, and mouth, you can possibly be infected with the virus. So we obviously wanted to develop a test or to test the fluid that is relevant to the spread of disease. The third reason is that different people shed wildly different amounts of the virus. So in a study released by Yale in April this year, they measured the amount of SARS-CoV-2 virus in an individual's saliva versus its nasopharyngeal swab. And what they showed here is that the amount of virus that they were able to measure in saliva is significantly higher compared to the amount that's found in the nasopharyngeal swab. Furthermore, they found that across different individuals in their study, the amount of SARS-CoV-2 measured here in copies per mil varies a lot from person to person. So some individuals can have about 10,000 viral copies per mil, but some other individuals can have about 10 billion or 1 billion viral copies per mil present in their saliva. So to be able to visualize this more, a person with a high viral copy number, about 10 billion copies per mil, represented here by person A, one tiny droplet from that individual is equivalent to about 5 liters of saliva from person B who only has 10,000 copies per mil of the virus. So no wonder we hear about super spreading events. So if you Google super spreading events or super spreaders, you'll find several cases happening in different regions of the world. And this is mainly because of super spreaders or people with high viral load. So it's very critical to avoid these super spreading events. So we have to test and isolate these people that carry a high viral load in their saliva. And so in late March this year, the governor of Illinois called for a shelter-in-place order, and that led the university to stop in-person classes. And they had to develop a system or a way for them to reopen in the fall semester. So Professors Martin Burke and Paul Garner, both from the Department of Chemistry, they were part of the COVID-19 steering committee that the Chancellor's office has tasked or assigned to develop a strategy for reopening in the fall semester. And so I am a postdoc in Dr. Hergenrother's lab, and he knows that I have a background in molecular biology. And then I was volunteering for the campus efforts to provide viral transport media to the state of Illinois. So he asked me about the current status of testing strategies back in April. And we addressed some of the bottlenecks that are encountered by these tests. And together we devised a plan to develop our own testing strategy on campus. So that's how the Illinois Shield program was born. So Paul then recruited other professors, Dr. Tim Tan from the Department of Vet Medicine, and also Dr. Crispr, a virologist, and all these other professors sent their postdocs and research technicians to be a part or assigned their postdocs and research technicians to be a part of the team that will develop the testing strategy for campus. And so we called this the Illinois Shield Target Test Intel. And as you can see here, the test part is in all caps, highlighting the importance of the testing strategy for this campus reopening. And so this project was completely funded by the University of Illinois-Urbana Vice Chancellor for Research Office. And so when we looked at the current status of SARS-CoV-2 testing strategies back in April and May, the gold standard has been using the nasopharyngeal swab placed in viral transport media And then using this specimen, RNA purification is performed to isolate the RNA of SARS-CoV-2 and then perform RT-PCR. However, both the nasopharyngeal swabs and VTMs are already in short supply back in May. And the RNA purification kits are also in short supply due to global demand. So in early April, Rutgers came out with their first FDA EUA approved saliva collection device. And they reported their success in using saliva as specimen. But in their protocol, they still perform RNA purification followed by RT-PCR. So for our campus, our goal was to develop this testing strategy using saliva and cutting out this RNA purification step to remove one of the major bottlenecks and then go straight direct to RT-PCR. So all of this is reported in our preprint that we released in mid-June this year. So why saliva? So we have several logistical considerations. Number one, we wanted to avoid swabs, CTM, and RNA extraction kits, all of which are supply chain limiting. We also want a test that's easy and is amenable to rapid self-collection. There's also no need for healthcare workers to collect the samples. And it's much easier for ambulatory patients or individuals to hand out their saliva samples. And using saliva as specimen is also compatible with frequent repeat testing. So it gives you a more uniform specimen over time in contrast to a nasopharyngeal swab specimen, which depends a lot on the skill of the healthcare worker that's collecting the sample. So the two figures shown here are our guiding principles for deciding on the frequency of our testing and the development of a speedy test. So looking at the pink bar, which corresponds to highly sensitive tests such as RT-PCR tests with a limit detection of 1,000 copies per ml. The purple bar corresponds to rapid antigen tests with a limit detection of 100,000 copies per ml. So we're looking at the frequency of testing versus the percentage of infectiousness removed, or your ability to control the spread of the virus. So if you don't do any testing, you obviously will not be able to limit the spread of the virus. If you do a daily testing, on the other hand, looking at the pink bar, you would be able to control the spread of the virus almost to 100%. However, we all know that a daily testing is not feasible because RT-PCR is an expensive test. And so we needed to decrease the number of the frequency of testing. And so in their simulated program, if you perform testing every 3 days, you are still able to control the spread of the virus at greater than 80%. If you do weekly testing, you're able to control the spread of infection to about 8%, and so forth. And here the figure on the right shows you what will happen if you turn in your results within the day or after 1 day or after 2 days. So focusing here on 3 days, every 3 days testing and focusing on the pink bar. So if you are able to return your results within the day or zero day, you would be as expected, you would be able to control the spread of the virus greater than 80%. However, using the same testing strategy, which is every 3 days, and using the same RT-PCR testing, if you give back your results after 1 day, you dramatically reduce your ability to control the infection down to about 70%. And it further drops down to about 50% if you turn in your results after 2 days. So obviously, the frequency and the turnaround time of the test highly matters. And that test sensitivity is only secondary to frequency and turnaround time for COVID-19 surveillance. So for scientific and medical considerations, we know that the spread of SARS-CoV-2 is through aerosol and droplets. So saliva is a more relevant media to test. And we also know that high viral loads in saliva can be measured during an individual's infectious phase. So the goal for testing development and application is to devise a test that bypasses RNA isolation and purification. So this would require the development of a new test. We also want this test to give us fast reporting of results. So ideally within 5 hours or up to about 12 hours return time. And this would require that this test site should be on campus. And we wanted a test that will enable us to screen about 40,000 students, faculty, and staff on a twice-a-week basis. So this would require us to develop a fast and efficient collection strategy. And all of this also suggests that we need to assign a high complexity laboratory where this lab will have the experience in manual molecular testing, and the test itself will be performed by highly skilled technicians. So to give you an overview on the SARS-CoV-2 biology, we know that this virus is an RNA virus, so its genetic material is made up of viral RNA held together by the nucleocapsid protein or the end protein. And this genetic material is encapsulated in an envelope that in itself also contains several different proteins such as the spike protein or S protein, the membrane protein, and the envelope protein. So the RT-PCR kit that we decided to use was the product that was developed by Thermo Fisher Scientific. So they came up with this COVID-19 combo kit. And this kit enables us to perform multiplex PCR. So meaning it will enable us to target multiple regions of that viral RNA. And this will increase the reliability of the test. So looking at this viral RNA sequence of SARS-CoV-2 RNA, it's made up of about 30,000 nucleotides. So the TAC COVID-19 combo kit will target a region of the ORF1 AV gene, the S gene, and the N gene. So using the reverse transcription quantitative PCR as our technique of choice. So to give you a basic principle of RT-PCR, so I mentioned to you before that the viral RNA or the genetic material is made up of RNA, so it needs to be converted to DNA through a process that we call reverse transcription. So that's the RT part. And once you have your DNA in solution, the primers and enzymes that are present in the mix will be able to amplify or copy this DNA or genetic material into several copies in about 40 cycles or so. So technically, in each cycle, you double the amount of your genetic material. So if you have one copy at the start, at the end of cycle 1, you'll have two copies. At the end of cycle 2, you'll have 2 to the 2 copies. So that's 4. And then it will exponentially grow in every cycle. So this is the exponential curve that you will generate looking at the number of cycle and the rest that's amplified in each reaction. And the key here for understanding RT-PCR results is that the CT value or cycle threshold value that's reported at the end of this experiment or at the end of this test is inversely proportional to the amount of DNA that's present in your starting material. So meaning if there are more viral copies present in saliva, the lower the CT value that you will see at the end of that assay. So we were told that failure is not an option. We know that already at the start of our project, and so we worked relentlessly to develop this test in a short amount of time. So we optimized several different conditions in order to come up with a simplified testing strategy. So we optimized the temperature, the time, as well as the buffer and additives that we added saliva. Determining the perfect conditions at which we could still detect the virus that's present in saliva. So we worked with a heat-inactivated virus, a gamma-irradiated SARS-CoV-2 that we used to spike known negative saliva samples at known concentrations of the virus, and then establishing the limit of detection. So we compared an approach at which we start with saliva, perform RNA extraction, and then RT-PCR. So that's the two plots on the left here. And then in contrast to a protocol in which you heat your saliva and then you perform direct RT-PCR and setting or determining the limit of detection for that approach compared to that one with an RNA extraction step. So focusing first on these first two plots on the left side. So we established the limit of detection. So LOD is established by performing serial dilution of known amounts of the virus. So starting with a high concentration, about 500,000 copies per mil, and then performing serial dilution of that virus down to about 100 copies per mil, and then running your performing RNA extraction and running your RT-PCR experiment. So the LOD is set at the amount or concentration at which you still detect all of your replicates in the assay. So for the protocol that performs an RNA extraction step, the LOD is set at about 500 copies per mil. So it doesn't matter if you don't add any if you add buffer to saliva, or if you don't add any buffer to saliva, your limit of detection will always be 500 copies per mil using this thermal pressure pat COVID-19 kit. And in contrast to that, if you cut out the RNA extraction part, so you have saliva spiked by the virus, and then you heat it for at 95 degrees for 30 minutes, and then add the buffer TBE, and then perform at a 1:1 dilution, and then perform RT-PCR experiment, the LOD would now be at 2,500 copies per mil. So when we slightly modified that experiment to add Twin 20, which is a detergent that we know will help open up the virus further. So if you add that detergent, your LOD now becomes 500 copies, 500 to 1,000 copies per mil. So it's very close to a protocol at which you use RNA extraction in your screening strategy. So this limit of detection, which we set at 1,000 copies per mil, is reproducible, as shown here on the left, where you spike several different saliva from different donors or from commercially available pooled saliva. If you spike it with 1,000 copies per mil and you try to detect that virus, you will be able to detect that a CT value of about 32 or in the range of 30 to 33 CP. And we also know that the test is specific to SARS-CoV-2 because you only detect your three target genes if you spike saliva with your known inactivated SARS-CoV-2, or it's also specific to if you just spike it with the N transcript of SARS-CoV-2, you only detect the N gene. And if you spike saliva with other known human coronaviruses such as the OC40 and N229E, you don't detect the three viral target genes. Similarly, if you use synthetic RNA constructs from SARS-CoV-1 or the original SARS virus, as well as the MERS-CoV coronavirus, you also do not detect the viral target genes. And HEK293 is RNA extracted from human cell lines, so you don't also detect the three genes using the RT-PCR kit from thermal pressure. So this suggests that the test is specific to SARS-CoV-2. So we came out with our protocol, our simplified protocol, which, as I mentioned before, is collect the saliva. You heat it at 95 degrees for 30 minutes. You add your TB buffer and Twin 20 at a 1:1 ratio, and then perform a direct RT-PCR assay. So in addition to our group, The Yale group was also able to develop their own direct saliva to RT-PCR assay. So how they differ from our protocol is just at this one initial step where, or at least two steps where you collect saliva, you add proteinase K to that saliva, you mix it, and then you heat inactivate it for 5 minutes, and then perform direct RT-PCR. So we compared the two different protocols and established the LOD of each of these respective protocols. So as you can see here, the plot on the top is following the UIUC protocol, and the plot on the bottom show you the LOD for the Yale protocol. So what this data shows is that the limit of detection for our UIUC protocol is right at 1,000 copies per mil, in contrast to 6,000 copies per mil following the Yale protocol. So everything else kept the same except for their initial processing approach. So using our protocol, an LOD of 1,000 copies per mil or 1 copy per microliter would mean that we will get a CT value of about 35 to 37. So is this relevant in the real world setting? So at least two papers have come out showing that the contagiousness of a person is inversely proportional to CT value. So what this French group has reported is that if a patient known to be positive for SARS-CoV-2 has a CT value of 13 or any low numbers from 13 to about 20, if you try to culture the virus from their saliva, you will be able to collect or 100% you will be able to isolate positive cultures from the sample. However, if you have a CT value of greater than 34 or 35, 36, and so on, they are no longer able to isolate positive cultures from the sample. The same findings were reported by a different group, this time from Oxford, and also in their hands, they showed that a CT value greater than 35 means that the virus present in the sample is no longer active or can no longer be cultured or isolated in culture. So patients with CT values of 35 and above are no longer contagious. So we also measured the stability of saliva at different storage conditions and determined that saliva is stable at room temperature for up to 7 days prior to heat inactivation. So the experiment here on the left involves spiking gamma-irradiated SARS-CoV-2 in saliva and storing it at either room temperature or at 4 degrees Celsius at different time points, and then measuring the presence of the three viral gene targets. So as you can see here, if you store saliva at room temperature prior to heat inactivation for up to 7 days or 168 hours, you still get the same CT values compared to a virus that has been freshly spiked saliva and processed right away, or that has been sitting at 4 hours or 6 days. So it's the same result. Or in fact, storing saliva at room temperature is better than storing it, or slightly better than storing it at 40 degrees Celsius. So the figure here on the right is data from an actual patient sample. So pooled clinical sample that's known to have or is positive for SARS-CoV-2. So if you store that saliva either at room temperature or 4 degrees or on ice or outside, with a temperature ranging from 23 degrees to 27 degrees Celsius back when we performed this experiment, what you can see is that the virus remains relatively stable at any of these conditions. So as long as we don't heat inactivate it, it will be able to stay for a while. The virus will be able to stay for a long time in saliva. So the next step is to, of course, obtain clinical samples and perform paired testing of nasopharyngeal swabs and saliva samples using our thermal pressure COVID-19 combo kit. So the experimental design is collecting from the same individual both nasopharyngeal swabs in the TM and also saliva. So the swabs in VTM will undergo RNA extraction steps prior to RT-PCR, whereas saliva protocol will go direct from saliva collection to heating and then to RT-PCR. So we obtained several patient samples from a local hospital drive-through COVID-19 testing clinic, and then we also obtained samples from inpatient individuals who are known to be infected or positive for SARS-CoV-2 and are confined in the hospital. We also had samples from the Champaign-Urbana Public Health District, as well as the University of Illinois Department of Division of Illinois Athletics. So for a total of 267 specimens collected. And then, as shown in the table here at the bottom, so out of the 33 positives that came out of the nasopharyngeal swabs, 30 samples were also positive for saliva. 3 samples came up negative in saliva but were found to be positive in the nasopharyngeal swab. However, when we tried to extract RNA from saliva, we didn't detect any of the viral target genes, so suggesting the difference between the nasopharyngeal swab and the saliva specimen. So overall, our positive percent agreement for the sensitivity of the assay is calculated at 19.9%, and our negative agreement where we were able to detect 234 negative samples out of 234, so that gives us 100% specificity for our assay. So this slide gives you a timeline of our assay development phase. So Dr. Garner and I started hatching plan in late April, around April 2022 to 2027. And by the end of that month, we were able to assemble our team who performed the experiments. And our first official day started last May 4th. And on that day, we didn't even have a formal lab to do our experiments on. So we had to look for a lab on campus that will enable us to perform COVID-19 experiments, get approval from the university in order to do some COVID-19 work, and also start working on the IRB protocol for obtaining for clinical samples at a later time point. So around that time, most of the labs on campus have already loaned their RT-PCR machine to local hospitals in town, so that those hospitals can perform COVID-19 testing in the community. So we had to look for other available RT-PCR machines. Fortunately, we were able to find 3 more qPCR machines on campus, and this is Paul, my boss, and my other teammate. So they picked up some of these RT-PCR machines to bring it to the lab that where we decided to perform our experiments at. And after 2 to 3 weeks, we already know that the protocol is sound, and that by the end of May, we already worked on setting up our high complexity laboratory on campus. And at the first week of June, we were able to obtain already clinical samples from the hospital. So this is me and my teammate Robin, and we were delivering 50 ml cultures to the different testing sites. So we were able to fit in 10,000 tubes in both cars. And so starting early June, we have been able to perform our clinical samples for testing clinical samples. And by mid-June, we already reported our saliva preprint paper, and you can find that bioarchive. And so at the first week of July, we already started testing faculty and staff on campus. So our capacity was around 1,000 tests per day. And then in mid-August, so about two weeks prior to the start of the fall semester, we started hiring more staff so that we're ready to perform about greater than 10,000 tests per day on campus. So we started screening students that have started moving back in on campus, and This is our team, and this is me holding up, doing our meetings in between shifts. So our team huddle, and then this is me performing or preparing the master mix and loading up our six RT-PCR machines. So that's how we're able to perform more than 10,000 tests per day. And in early September, I was contacted by Senator Gordon of the Philippine Red Cross, stating their intent to use our protocol and combine that with their existing labs or protocol in their labs. So they already have their own RT. They're using their own RT-PCR kit from Sancher, and they just needed to perform validation tests to show that our upstream protocol can be used to perform direct RT-PCR from saliva using the Sancher RT-PCR kit. And so on September 18, an undisclosure agreement was set up between UIUC and Philippine Red Cross, and also in collaboration with several professors from UP Manila. And on September 25th and October 7th, we presented the findings to the Department of Health. So we're seeking, or the Philippine Red Cross and UP Manila team are seeking for approval from DOH to allow the use of saliva as a specimen, and also allow them to perform direct RT-PCR. So just to give you an overview of our current testing metrics. So today, since we started in July, we have already performed greater than 900,000 samples or tests. And currently, our 7-day case positivity rate has been largely maintained at 0.33%. So it's less than 0.55%, which is a pretty good number compared to what I told you earlier, where the percent positivity rate in the state of Illinois is about 12%. So we did experience a big spike in late August, and this coincided with the time at which students started moving back in. So a lot of them tested positive, and so we needed and doing the tests on a consistent basis allowed us to identify those positive individuals and isolate them right away from the community, and that's how we have been able to relatively control the spread of the virus on campus. So Illinois Shield, as you know, is target test and tell. I told you mostly about the test part, and the tell part is about the app that was also developed on campus. So this is the Safer Illinois app that everyone on campus is required to download. And so this is tied up with our testing results. So an example here is what's shown for my Safer Illinois app. So it will reflect that I went through a recent testing and that I tested negative, and so I am allowed building access. So what's the next step beyond UOI? So I told you about the 10,000 to 15,000 tests per day performed at our campus testing facility. So right now we're also getting ready to launch our Shield Annex, which is made up of 5 mobile labs. Each of these mobile trucks are able to perform 10,000 tests per day. So we have a total of 5 mobile trucks or mobile labs, and so that means we'll be able to process 50,000 tests per day. And the Shield Annex is designed to help the original testing lab with performing tests to the Champaign-Urbana community. So not just the campus community, but also to those residents outside campus. So within the cities of Champaign and Urbana. So this is a picture of one of our Shield mobile trucks, and so each of them is fully equipped with all the equipment and reagents required to perform the test. And Last but not least, we also have the Illinois Shield. And for this part, we are working with the Illinois governor to expand our reach to the entire state of Illinois. So we aim to open up 7 more labs by the end of 2020 and 3 more labs in 2021. So that will give us a greater than 70,000 tests per day capacity. So all in all, we want to target up to 150,000 tests per day or 4 million tests per month. And we are currently in discussion with many potential national and international players. And eventually, we would want to move on to a point-of-care test because obviously this is a very expensive endeavor. So several other laboratories on campus are currently working on developing a faster and cheaper point-of-care test. So those are your rapid antigen tests. And so to summarize, so I told you about the simple protocol that we developed that cut out or remove all the supply chain limiting reagents. So this allowed us to develop a relatively inexpensive and frequent testing. So our testing process was already demonstrated on campus and over 900,000 tests so far and counting. So we're rapidly approaching a million tests performed since we started in July. And we also have rapid results reporting, which enabled us to perform rapid contact tracing. And the test is performed on specimen that is most relevant for the disease spread. And with that, I would like to thank you for listening, and I wish you to stay healthy, safe, and connected. And please, let's work together to get saliva testing to the community. Thanks again, and I'm open to your questions.
Okay, thank you, Dr. Diana Ranoa, for that very interesting lecture, for sharing with us your what's keeping you busy there at the University of Illinois. So this is indeed another advancement in COVID diagnosis. No? So saliva testing will make it easier for patients to get tested and for us physicians to diagnose them early. So we have a lot of questions in the Q&A box, but unfortunately, we'll only be able to entertain a few of them in the interest of time. So first question, what is the turnaround time of the test from saliva collection to the availability of the result?
So yeah, it depends on the number of samples we receive per day. So if it's at greater than 10,000 samples per day, our turnaround time is about 12 hours, 8 to 12 hours. And we actually categorize our samples. We have some priority racks. So those samples or those individuals can get their results as quickly as 5 hours.
Okay. So can we use the currently existing RT-PCR machines that we use for nasopharyngeal swabs for saliva specimens?
Absolutely, yeah. So in fact, when in my collaboration with Red Cross, they're using their own Sansure machines. So they also use their own Sansure kit. So they just did some validation tests using the upstream part of the protocol. So definitely the technique can be applied to several different types of kits and PCR machines.
With regards to the timing of the test, is there a specific time where you get the best result, like timing in terms of the illness?
So to answer that question, in terms of illness, we don't really account for that because we're regularly testing everyone twice a week. So we easily catch infected individuals. And then for the day, like it doesn't really matter when you go to the testing site to give your saliva sample. What we require from people is that they don't eat or drink within an hour, 30 minutes to 1 hour, just to make sure that there are not a lot of active enzymes that could potentially degrade the RNA in saliva and give you false negative results.
I see. We have your question from Dr. Buensalido. So he says, congratulations. And the question is, have you encountered variations in the CT values from different RT-PCR machines or brands?
Yes, so that's why it's important to establish the limit of detection on a PCR kit basis. So yeah, what I showed you today is just for the Thermo Fisher TaqPath kit, which gives you an LOD of 1,000 copies per mil. But say if you use the CDC primers, CDC recommended primers, and the TaqPath master mix, it will give you an LOD of about 2,500 copies per mil. So definitely you'll have to check what the LOD is for each kit.
Okay. For our next question from Dr. Katrowa, can you estimate in Philippine pesos how much each test would cost?
Yeah, so because we were able to cut down the reagents and a lot of supplies, we've calculated it to be around 18 to 20 US dollars, or roughly around 1,000 pesos, using the Thermo Fisher TaqPath kit. So that one's an expensive kit, so it will definitely vary. The price will definitely vary depending on what kit you're using. So I would say it's 1,000 pesos. And I think after talking to Red Cross, they also said that their calculated cost, if they cut out their RNA extraction part, is also between 1,000 to 2,000 pesos, down from 4,000 pesos.
Oh, okay. So that is indeed much cheaper than the currently being used NPS, OPS for PCR. So, have you encountered any cross reactions with previously infected patients for the test?
None that I know of. I'm not privy to that information because we have HIPAA rules, so we only test and we don't really see if it's a patient who already contracted the virus before. So I can't answer that question.
Mm-hmm. And maybe for our last question, is this method also applicable to pooled testing of saliva?
Yes, it is, but it's not advisable to do it when the case positivity rate is high. And also when we did the batch testing, at every dilution that you do, you lose one CT value. And also at the rate that we're doing the tests at 10,000 tests per day, it's not very time efficient for us to pull the samples because it's complicated to backtrack. The matrix is going to be complicated if you end up having a positive sample and you have to trace back again from that pool. Yeah. So we decided to just not do any pool testing. But yes, it's definitely amenable to that technique.
All right. So in the interest of time, we have to end this session. It's already 9:14. But we would like to award to Dr. Diana Renoa the Certificate of Appreciation. So I won't read the whole certificate, but we would like to thank you for sharing with us your expertise and your latest project in the University of Illinois during the 42nd Annual Convention of the Philippine Society for Microbiology and Infectious Diseases. And we look forward to a time when saliva testing will be widely available and it will be easier for patients and physicians to diagnose COVID patients.
Thank you for having
me. With that,
me.
Thank you, Diana. And with that, I close this session. And we invite you tomorrow to join us for the Antimicrobial Stewardship Workshop, which will start at 10 a.m. So thank you, everyone, for joining us, and have a good evening.